So those who will be starting first, second, and third. Like that in that order. So please ensure that you have your presentation already sent with us. Before that, of course, we are going to have our guest speaker who will open, and then after that, you will follow in the order in which you appear in the program. Thank you. Madam Baye. Madam Cecilia.
recap. My name is Lord Abe, and I'm a professor in Dalhousie University and horticulture, so I'm an agricultural scientist. Okay, so I would like to talk about understanding resilience in a complex systems. So if I say complex system, we all know how climate change and food insecurity, nutrition insecurity, and societal health and so on and so forth, are impacted either by our making or unmaking. So I want to discuss that, and based on that, I will try to link this topic with the theme for the conference, okay? And we all know about the theme of the conference, so I'd like to link that to the theme of the conference. And this meeting, those of you who were in the presentation, the last keynote speaker talked a lot about the theme for the conference. So I'd like to link my presentation to that. So what you basically see is a brief talk on the theme, and then also some research that I conduct in my lab that can be linked to the theme. I think on Thursday too I'm speaking, so Thursday to be more scientific topics on more on details on research. Okay, so as we all know, we can't overemphasize. We all know about the climate change and how that has impacted food and nutrition insecurity, and for that matter, technological disruption are no longer distant risk because we experience it each day, right? And across various parts of Africa, so moving from coast to coast to coast, we see how this impact is occurring: rising temperatures, unpredictable rains, and then declining productivity and yields, and even declining food quality. We can see that and feel that. So this is not an isolated challenge, but if you think about it, how climate impacts agriculture, you can see that it impacts our water systems too, because heat stress, drought, etc., etc. So it impacts our water systems. It impacts livelihoods, of course, livelihoods of farm households, livelihoods of society, because people depend on agriculture. And as you know, in Africa, agriculture is is a great contributor to our health, economy, and so on and so forth. So this is very important. Because of these challenges, we see farmers adopting something like hydroponics, right? Because they need to create, they need to think outside the box. So we see local farmers trying to adopt something like hydroponic systems. We see research says, developing heat-tolerant crops, and this is very important because we need to, develop crops that will adapt to the climate change and to what's happening in our environment, the ecosystem. We see young entrepreneurs building vertical farms in dense urban spaces. So we have rooftop gardens and so on and so forth. I don't know if you have rooftop gardens in Kenya, but it's a growing thing in so many places in Africa, including Ghana, where I come from. So this is what it means to reimagine tomorrow, as the theme says. So as I've said, you can see that because all these things are interlinked, the challenges we face is deeply interconnected. Okay? And as such, we need that kind of interdisciplinary thinking to be able to move forward with this. It also means that incremental change is no longer enough. And therefore, we must move from fixing isolated problems. We see those problems being isolated. We must try to move away from that to redesigning the entire systems. And that is what farmers, local farmers are doing. They are not researchers, they are not scientists. I think I will remove my jacket and sweat. Okay, so we see researchers and farmers trying to think outside the box. Okay, so this is ongoing. So, and the keynote speaker, as I referred to, also mentioned about silos. We must break down those silos. So one of the greatest barriers to our progress is how knowledge is organized in silos. So you see horticulturists doing this own thing, you see engineering doing their own thing, and so forth. So we must break that. Instead of working in silos, we must work as interdisciplinary teams. Thank you very much. Okay. So example is researchers and farmers work together on agroforestry. And we know what agroforestry is. It's integration of tree crops or trees and then fruit crops. So by doing this, they diversify. And when they diversify, you have your trees, you have your probably cover crops, and so on. So if you're able to improve soil health, they're able to diversify. So more crops, if one fails, the other will survive. They're also able to improve and diversify the revenue, farm revenue, okay, and also the nutrition of people. So if you are doing this as a farm household and you are doing agroforestry, you are able to integrate leguminous crops to tree crops and so on and so forth. So you may have your cash crops there and you may have your food crops there. So we can do that. So we see farmers doing this, and it's a big thing in so many places in Africa, agroforestry. So this is not just agriculture, but ecology. It's ecology because we see in the soil health, environment is improved, the microenvironment is improved, soil health is improved, so ecology is there, we see nutrition there, and also the economics because of the diversification. So this is the power of interdisciplinary thinking. We always have to think outside the box. And how we can think effectively outside the box is by doing interdisciplinary research. So for long, we think of resilience as the ability to bounce back. Okay, so if you say someone is resilient, probably the person went down and the person came up, so bouncing back. So that's how we think about resilience. But we can redefine resilience. We can redefine it as the ability to adapt, evolve, and thrive under uncertainty. And this is what we see now. I talk about the hydroponics, I talk about agroforestry, and so on and so forth. So this is farmers and researchers thinking about how to adapt to the situation. Okay? If it's climate change, soil health going down, and so on and so forth, how to adapt to that problem, how to come out of that problem, and then try to move on. And I gave an example here as where rising sea levels. So due to the climate change, we see it's happening everywhere. Due to rising sea temperatures, we see ice melting, rising sea level, even in Canada. Coastal regions are suffering because of the rising sea levels, which is infiltrating into the soil, the surrounding soils. So soils are becoming more salty, they're becoming more saline, and it's not supporting production. So more research is being done to bring up either to improve crops or to select varieties that can tolerate the salt. So trying to think outside the box, as I've said already, and also to be resilient against that, so adapting to the change. So resilience is that not returning to the past, but preparing for the future. Okay? So that's why resilience means not returning to the past. So we can't say bouncing back. So bouncing back means you are returning to the past, but we want to move forward. We want to look at what happens in the future. That's what we want to prepare for. So that's what resilience means. So based on this, my research focus, I want to talk about a bit of my research. My research focus is based on health-based sustainable food systems and food security. And it's highly interdisciplinary and multidisciplinary as well. So I try to collaborate with people from different disciplines. I do research with engineers, I do people in medicine are doing research with people in nutrition, food scientists, soil scientists, and so on and so forth. It's highly interdisciplinary. And within my lab, I have my postdocs there, I have my PhDs and masters and so on and so forth. And I try to recruit people with different backgrounds, from different backgrounds, so that they will help have that multidisciplinary, or multidiscipline in my lab. And that helps a lot, trying to, work as an interdisciplinary team and multidisciplinary team as well. I try to make the two because I think having the two is very powerful. So we have, so my research, if you go on our website, my research is mainly on soil nutrients. So we look at soil health, we look at improving soil, and either in the field or in the greenhouses, so growing media and so on and so forth. We also look at plant response to biotic and abiotic stress factors. We also look at greenhouse technology and production systems. We look at value addition to compost. I work a lot on compost and compost utilization. And then valorization of biomass. So valorization of organic products. How do you convert organic products to either food or to amendments or to biostimulant to help production and also help with plant health. So when we talk about plants, we have to look at plants as a whole, okay? Right from the soil, we look at the roots, the stem, the leaf tissue, the flowers, the fruit, whatever it produces, if it's fruit, the fruits, and so on and so forth. So we have to have a holistic view. So climate change impact on plant, it doesn't affect only the soil, but whatever affects the soil also affects the plant and affects plant health and productivity. So we have to have that, thought or thinking when we are approaching some of these problems, when we want to find solutions to some of these problems. And for that matter, as I said, I work a lot on soils and compost and so on and so forth. We have to, I believe that anything we do, we have to think about the soil first. And with soils, you can see here, humus, okay? Soil has colloidal particles, as we all know, scientists here know. It has colloidal particles, and the colloidal particles, is the mainstay of the soil, okay? That determines the soil health. So this is core, it's very, very important, it's very critical. But people, animals, and plants, okay, we interact in such a way that we impact the humus, the soil health. We impact it a lot, either through cultivation, tillage of the soil, or through waste that we generate, the residues and so on and so forth. And that impacts the soil, that impacts the biological properties of the soil. So we look at the soil fauna, the soil fauna, and then the plant roots and microbes, like the soil microbial population. We impact it a lot in what we do, whatever we do. Also the physical properties, so the soil structure, aeration, and so on and so forth, we also impact that a lot. And also the soil chemistry. So the pH of the soil, the fertility, so the anions and cations, we also impact that a lot. And all these affect the humus, as I've said already, and then the active carbon content of the soil, which holds the nutrient and releases the nutrient whenever the plant needs it. It impacts that a lot, and that affects plant growth, plant health, etc., etc. So, thinking about that or about this concept, helps you sort of redefine your research problems and what you think have to be done to help to the plant. So one of the work that we did is compost residual. So we apply compost to the soil. We did this work, it's a 5-year research, and we did it in a province called Manitoba in Canada. And the test plants were lettuce, carrot, beets, and green beans. And we used a soil that is highly exhausted, so it's very poor in nutrient content. And we were applying normally in in the papers and recommendation, they will say apply 40 tons of compost per hectare or 20 tons and so forth, large tonnage to soils. But what we did was that because it's a 5-year continuous work, we're applying just 2 tons per hectare to the per hectare to the soil, just 2 tons out of compared to the recommendation of 20 to 40 tons. So we did this over 5 years. And at the end of the 5 years, what we did was that, that's the 6th year, we took some of the soil samples. I brought it from Manitoba to my lab in Nova Scotia. So we have to fly it, we have to ship it there. So we brought some, store it in the fridge. We have a walk-in fridge. And then I did some potted experiment just to see the residue. So after applying these 2 tons, just 2 tons over 5 years, what happens to the soil health characteristics? Because sometimes farmers complain, we can't have access to compost, or we don't have so much compost to apply, and so forth. But what I keep on telling them based on this, that don't think about having large quantity. And even in Kenya here, last time when we came, we visited some farms, and they were also saying similar thing, the source of compost, the quantity to apply per acre or something. But what I keep on telling them, don't worry about how much you need to apply, because even if today you got your 40 tons and you apply, plants will not respond this year. They will respond maybe 2-3 years. That's when they will benefit from the compost, because compost is a slow release. So applying in small quantities is just enough. So we did that, and then we look at some parameters. We have published about 3 papers out of this. So this is just a few of the data that I'm presenting. So we look at the nitrogen, the carbon, the active carbon, and also CN ratio, and then the biological nitrogen availability, which is nitrogen mineralization, what is bioavailable to the plants. And you can see the control. In the control, we did not apply compost at all. And then in the annual, we apply every year. So every year we apply 2 tons. And then in the biannual, we apply every two years. And you can see when you apply 2 tons every year, just little amounts every year, you can see how the nitrogen builds up, right? It builds up the carbon, the active, the active carbon, which is very, very essential. That's the active part of the organic matter, which is very essential to the soil. If you want to detect soil health, the active carbon is very important. The CN ratio, you can see that the CN ratio went down because of the buildup of nitrogen. So the CN ratio went down, which is great. And then the biological nitrogen availability also went up, twice the biennial, and then more than 4 times the, compared to the control. So it built up significantly. And you can tell from the field, when you walk into the field, you can tell from the color of the field. You can see the control is very pale, and then you can see the annual application very dark, and then followed by the biennial. So it's visible. From the 6, 7, 8 years going, it was so visible. And I asked the farmer, don't apply anything, just plant. And the farmer was planting and was getting more. So the residue worked, right? So farmers can actually, apply whatever they get, apply. Even if you are applying fertilizer, still apply the compost, because compost helps in so many ways. So this is what we found, which is very encouraging. Then we took the plant too, and then we did some comparison. And you can see the annual. This is the size of the different sizes. If you look at the beets, you can see, compare the lettuce, then the beans, and then the carrot. Very, very significant. The difference is so obvious. So, and if you walk to the field too, you can tell from the chlorophyll concentration of the different plants. So application of compost helps. Now, we also look at more parameters. We want to look at the food quality. So we did some biochemical analysis of the food, looking at something like succinic acid, glutaric acid, and so forth. And these are all tablets. These compounds, you can find them in tablets that are given for people to take, because sometimes people are sick, cancer, and all those things. And they are given these tablets to take. And the annual application applying this small compost, in fact, builds these compounds in the plants. So compost is great. I was telling the Compost Council of Canada, they invited me to a talk, and I was telling them that we can even, I would, if I'm not being too bad or terrible, I would say that we should produce compost tea for people to drink. I was just joking. But you could tell how healthy it is, right? Apply compost, which people don't consider important. So you can see, and then we look at the soil microbial enzymes. So the enzymes that the soil microbia produce to help improve and enhance the soil health, you can see it's also increased. How can you tell? You can tell from, if you look at the AN is the annual application, and then AN beets, and then AN green, that's the annual applications. And then when you see the CB, these are the controls. So this is the annual application. You can see that all the annual applications are gathered in these quadrants, right? So these compounds are close to that. It means the annual application enhances those compounds in the plants. And then you can see where the control is. The control is far removed from all the compounds. So it means there was nothing in the control plant. The control plant did not enhance those compounds in the plant. So compost actually helps. And you can see the BI. BI is the biennial. You can see where the BI lies. So it's somewhere in the middle. So this confirms that the compost actually help improve these compounds in food that you eat. Okay. Then the soil that I took, that's my hand by the way. Just to confirm that the work is real. Yes, I don't want to play all, so. Okay, so. Okay. I tried this and it worked. Let me try that and see. I'll try it. But I'll try this and work. Otherwise, I won't. So so what we did was also we took some of the soil that I brought in my lab, and then we tried to look at the how resilient or how healthy the plant has become. So we imposed stress on the plant. So we grew these tomato plants. They are the same cultivars. They were started from seeds at the same time in the same environment. They were treated the same, and they were transplanted the same in the same amount of soil, the same type of pot, and then put in a place in the growth chamber. So we allow it to grow for about 3 to 4 weeks after transplanting, and after transplanting, you can see the color. You can see that this one is pale. This is the annual application, and this is the control. So after allowing them to grow for 3 to 4 months, then we stop watering the plant. So we stopped watering the plant for about 2 weeks, no watering at all. So they were all, they became like this. So if you look at that, you can see that this is healthy well. It's growing good. The chlorophyll content is high, and it's more healthier. So it kind of has developed that resilience in it. It's more healthier. So you can see even after after 2 weeks or so without water, you can see in the small pot. So they were really, really stressed, and the soil water moisture content was about 6%. So it was quite dry, very, very dry. And you can see it's still standing, even though it's wilted, but it's still standing. And you can see this, so how it appears. You can see the control is completely, it's not permanently wilted, but it's wilted, to a very high extent. So what I did was that we want to see the resilience, so we added water to it. Oh, you were right. Okay, I'll start from here. So we added water to it to see their recovery rate. So just watch the recovery rate. And the recovery rate is based on the resilience of the Brazil Dreams plane. Yeah. So you can see, and we had the camera there. for 24 hours to see, and you can see how they are recovering. So this is a real-time recovery of the plants. So, as I said, we have this control, no compost, every 2 years, every year, and then this one was compost. That has stayed for about 6 years. And you can see the recovery. See, the one with the compost is recovering faster than the annual, which makes sense. And then if you look at the biennial, it's also recovering, but very slow rate, at a very, very slow rate. So this one recovered, this one recovered, these leaves are dead, they are gone, so that's fine, because they are older leaves. So they are old, so the stress will shock them. And then if you look at the biennial, it recovered, but not as much. But it recovered. After 24 hours, it was standing. But the older leaves were all gone. It remained with about 4 leaves. And the control was still looking like that. So this tells you that compost, can impact the plant's resilience and the plant health. So what we see in Africa, in warm climate and all that, we can help the farmers, or we can help the plant by applying compost to the soil. That's the takeaway message. We apply compost to the soil, enhance soil health. It will enhance resilience of the plants. So compost is very important. So what I say is that even if you have a plant, we are not asking farmers to produce organically, because in my belief, organic production cannot feed the world. That's what I believe. It cannot feed the world. I don't know, you may contest. I do a lot of organic research, but that's what I believe. So if we are applying fertilizer, apply compost and cut back on the use of fertilizer. You can cut back on the use of fertilizer by about 75%. So some fertilizer to help the plant, because of the population we need more. Because organic production has its challenges, a lot of challenges when you're producing organically. A lot of challenges. Weed control, pest control, disease control, and so on and so forth. So trying to meet the food need of growing population will become challenging. And when you're doing organic production too, it's usually not on a large scale. So what I say is that if you are producing using fertilizer, go on, but cut back completely, and then add compost to the soil. So the next experiment that we also did was screening of tomato, and I'm doing this in collaboration with scientists in Ghana. So they have bred some tomato varieties and so on, and then I had the seeds in Canada, and we are working on it. So these are the varieties that they have, and the Scotia variety is Canadian variety. Is stress tolerance, it's drought tolerance, and it's also flood tolerance. So we're trying to compare to see how best that does compared to the other varieties from Ghana. So the dark blue or the light blue are regular watering. So again, this was under water stress. So we have regular watering bars, and then we have the dark blue as flooding, and then the brown for drought. And you can see that in all the varieties, responded to stress. So we look at root volume, we look at root length, we look at so many parameters. This paper has been accepted this year for publication in the Canadian Journal of Plant Science. So so a lot of data, but I decided to choose about just 4. So we have the root volume, and as you can see, the regular watering has larger root volume, which is understandable because it's growing in a very good environment. And then, of course, we have the root length and so on and so forth. But you can see that all the varieties responded differently to the stress, to the drought, and then to the drought, and then to the flood. And the Canadian variety is doing well. In the we should look at that CV6 is doing well compared to that. It's comparable. And if you look at the Rootland to say. Then we look at the drought tolerance index. So the drought tolerance index, you can see that most of the Ghanaian varieties, well, not most of them, but at least 4 of them are doing very well. The CV1, CV5, and then CV7 and CV8, but CV6 is also doing well. The same thing as the flood tolerance varieties. In Ghana, tomato is a precious crop for us. We import a lot from Burkina Faso. So having this work is good for the country. So we try to compare and then see if we can also have a variety that we can use in Canada. But I guess the Canadian variety is also doing well, which is the CV6. So we can stick to our variety. But this work is good so that important, those crops that are more tolerant can be selected for Ghana and for any other African country who is also interested. So all this work that I do, is interdisciplinary. That's what I want you to keep in mind. The other work that we do, in combination with some engineers and some biochemists and all that, is biomass-derived biostimulant. And this work, some of the merit students who come to my lab, I've worked on some of these, and I've worked on this, like Martha Dabo, yeah. She worked on this, undergraduate student, she did a great job, and she's going to publish about 2 papers from this. Because the work that she did, she added maybe 6 months or so to be a master's degree work within the House standard. So we work on this, and I'm not going to disclose the details because of IP issue. So I'm not going to disclose the details, but we took biomass, it's marine biomass, and then we applied nanotechnology for some biochemistry, etc., etc. And then we produced biostimulant, and then we applied just to sort of increase or improve, enhance plant health and resilience and productivity, etc., etc. So we use different biomass to do this. We've gone on to even to use biomass and compost and some other plant materials and so on, and even fish waste and crab and all that. So in my Thursday presentation, you'll see more on some of this research. So this is great, and the formulation that we produce, so these are some of the interesting results that we have. And that's why I said farmers, I'm not saying advocating for stop using fertilizer, I'm advocating for cutback and use more organic cutback. So you can see this is lettuce and this is fertilizer. So this is the recommendation rate for fertilizer. We apply the recommendation rate, and then here no fertilizer. So 100% the recommendation rate, 75% the recommendation, 50, 25, and then 0. So this same recommendation, we applied the biostimulant that we derived. We applied 100% of the biostimulant, and we got this yield. So you can see how dramatic the yield increased. We applied 75%, and then we applied our biostimulant. Look at the yield. It's still more than the fertilizer recommendation. We cut back fertilizer 50% and applied our biostimulant. And our biostimulant, we apply 5 mils per liter. It's liquid, so 5 mils of our biostimulant, 1 liter of water, and mix. And if you look at that, 50% is still beat the recommended fertilizer rate. We applied only 25% of the fertilizer and applied our biostimulant. It still beat the recommended rate. Then here, no fertilizer. We applied our biostimulant. Because our biostimulant has some nitrogen in it, it's comparable to the 100% fertilizer rate. So the question is, if you have this biostimulant, why should you apply too much fertilizer, which it's not sustainable and can impact the environment, and even the food quality is not good. Because we have done a lot of work, and we published this in International Food Research Journal, to look at food, applying compost and some biostimulants, and look at the food quality, the nutritional impact and all that, and it's very high. In fact, organic source of fertilizer, impact the quality of the food a lot, and we have to think about that. So we apply that, and these are the results we have. And since then, okay, let me show you the, so the same work, this time in a graphical form, that's the letters, and these are the bars to show you more, the quantitative the outcome. So here we're able to, so fertilizer 100%, as I showed you, we're able to increase the yield by 28%, and then even at 0, we still had good yield compared to this. You can see that 160%. So it's a very good stuff, and we have an industry partner who is interested, and Larry is eaten widely in some African countries, including Ghana. Ghana is eaten widely, and when it rains, you can harvest it from the wild or around the house. It grows by itself, so you can harvest it. But of late, because of the climate change, it's getting endangered, quote. So people have started cultivating. But growing up, I didn't see it being cultivated. Even when you are eating this, think it's for the poor. But now, even the rich can't even get it to buy. And the work has been done on it, and it's found that it's rich in calcium, zinc, and so it's rich mainly in calcium is macronutrient, right? It's rich in micronutrient, and also some vitamin C and E, and it's rich in dietary fiber, and so forth. And it also has medicinal properties due to its richness in flavonoids, alkaloids, and some organic acids, and so forth. So it's used a lot as medicine, as food and medicine. After all, we say food is medicine, right? So it's a great vegetable that I'm trying to work on to understand it a bit better. So sustainable production is always a key to safety and profitability. I'm saying this because that same crop is known to be able to accumulate heavy metals. So if it accumulates heavy metals, that's also not good for health, right? So it depends on where it's grown. So we need, when it's grown, we have to think about this safety and sustainability as well. So we did some greenhouse work, and this work was done by, again, Mary student, Tabitha. Tabitha was in my lab for 6 months, and co-supervised by Dr. Peter Masindi. And yeah, she did great work. She has published one paper. She's preparing a second one for publication. I think it was submitted, second paper, or about to be submitted. So she did great work, and she was looking at nitrogen effect on the plant, and also looking at azomite. So azomite is a volcanic mineral, so it's mined. So she was looking at that to combine both nitrogen and azomite to see how that impacts productivity and then the quality of the food, because azomite is natural, so we expect it and micronutrient rich, so we expect it to enrich the micronutrients of the crop and then make it more palatable, nutritious, and so on and so forth. Yeah, so we did that work, and so few of the results here. So so you can see in the first set of photos, we have the 0 nitrogen, 50 nitrogen, 100 nitrogen, 150, and so on. This is great results. So it means that we can fine tune between 50 kilograms per hectare, 50 kilograms per hectare nitrogen, and then 100 kilograms per hectare nitrogen. That's the sweet spot. So, and this region is great. You can see the leaves, you can see the leaf size, the color of the leaves, and so forth. And then we came to, so that's first experiment 1. So after finding this, then we said, okay, let's add azomite to see what happens to it. Then we can also look at the biochemical composition. So we did that, and then we had 0 plus azomite, and then you can see 250 kilograms N per hectare plus azomite, and you can tell the difference. Again, the sweet spot is still here, right? But if you look at it, adding the azomite even enhances it more, the more. And she did some biochemical analysis looking at the health aspects, and it enhanced the health of the crop. So it's great, it's very interesting. And then she did a fresh weight. Again, you can see the fresh weight. Again, as I've said already, the 50 we saw in the photo, the 50 kilograms and then 100 kilograms plus azomite did the job very nice, especially at the 50. So it means, why would you add more fertilizer, right? Why would you add more fertilizer? Cut back fertilizer, add organic material, and then you get what you want. That's it. That's all we need to do. Okay? So it's proven here too. Then again, we did, I think this, okay, well, this is just to show the relationship, but it's still the same. You can see that the 50 kilogram and all the variables were there. So stem diameter, dry weight, and the fine details, you can see that they're all here. And also FBFM, this is electron transfer in the plants, in the leaves, the chloroplast. It's a measure of plant stress. You can see that where it lies very fine around this quadrant. So applying azomite and reducing nitrogen works. If you look at the 250, it's far remote from all the parameters, which means it did not help at all. Look at the 250, look at the 250 here, 200, 200, it did not help at all. So it's waste. So they rather stress the plant because they are far from the FBFM. So cut back. That's what we have to think about when we are talking to our farmers, and add compost or any organic source. So this work done by Tabitha will be continued by Betty. Betty is here too. Yeah, it will be continued by Betty. We want to look at the food quality and so on and so forth. So Betty is coming to work on that in my lab too, so that we can look at the quality now. Dive into the quality of the food, of water leaf as food. Then the next work is that we also did the response to Artemisia. I don't know if you've heard of Artemisia, but Artemisia is a magic plant. I came to a conference last year in Tanzania to present on this particular paper. It's a magic plant. Artemisia is grown in the wild. They've started cultivation of that plant. And it's good for malaria treatment. It's used in so many, I said, drinkers, so many things. And it's good for malaria treatment. So what I want to do, I want to, and it's grown in Canada too, but as ornamental plants. So we don't actually use it for anything. So I wanted to see if we grow this in Africa, and it's helping. What if we are going in a place like Kenya, around Mount Kenya, where it's cold, right? And places where it's hot and all that. How do we compare? What would be the artemisinin, the compound that is the active compound for treatment of malaria is artemisinin in this plant. So would that impact plant growth and then that compound? So we grow this plant and then put it in the growth chamber at different temperature regimes, 18 degrees, which is quite cold, and then 24 degrees and then 30 degrees. So after doing that, this is the plant growth. You can see that at 18, the plant is quite stressed, so it's palish in color. The internal two are very short. And then if you look at the 24, you can see that it's growing very well. It looks like the plant is very happy. And then if you look at the 30, it's growing well, but the plant was taller and the leaves were not large, kind of thing. So we also look at the top and down view of the plant, and you can confirm that the 24 really was helping the plant to grow very healthy. And even the 18 degrees, the leaves were larger but shorter. And then the 30 degrees, the plant was growing taller, but the leaves were not, they were like spines, or something like that. They were not big. So we look at some genes, some gene expression in the plant, and to see that gene expression is affected. So we harvested the leaves at different temperatures to look at gene expression. So we have ADS, that's the long name, and then we have this G2, the cytochrome, to see what's happening. You can see that it varies for the different temperatures, the gene expression varies. But what is more important is this one, the aura. The aura is a gene that, when it's expressed, more atomicity is produced. So it's related to atomicity, the aura. And you can see that if you look at growing, if you look at it, when we grow it and then harvest it, it gives at 0, 24 hours, 48 hours, 72 hours. You can see that the 0, we didn't see anything, okay? But immediately we put it in the code, so at 0, so that 0 means before we put it in the growth chamber at these different temperatures, we harvested the leaves and analyze the enzymes, the gene expression is 0. Then immediately we subject it to, we put them in the growth chamber. After 24 hours, you can see that the 18 is still 0. 24 hours, there was high gene expression. Okay, so the atomicity, what is telling us that atomicity increased after 24 hours, and then after 72 hours, it's quite low. So it's hit 24 hours and then dropped again, because probably it might have adjusted itself. Then if you look at the 48 hours again, there's variation. After 48 hours, that's when we had the gene expressed in the 18 degrees Celsius and so on. And then the 72 hours, it means when you started them to the different temperature, you have to wait a bit longer to see a real effect of gene expression on atomicity in the 18 degrees Celsius. And then followed by the 24 degrees Celsius and then the 30 degrees Celsius. So when you are doing atomicity, depending on the period, the time period, atomicity will be expressed differently. That's what this is telling us. Yeah. So so this is great work, and we intend following up and then studying more details into the atomicity and seeing how it works on different problems, maybe malaria and other things. But work is being done, a lot of work is being done in France and also Tanzania. They're doing a lot of work there. So at the conference they came to present, but I didn't see anyone from Kenya and other parts of Africa. But this is very useful. This plan will be very useful to us. So it's good that we also pay attention to that. So basically, that's the presentation for today, I guess. I was given 30 minutes, but I don't intend doing 30 minutes. So that's that. So as we leave this conference, I invite each one to ask questions. What systems can we rethink? And what disciplines can we connect together for us to achieve our aims and objectives, our goals? And what partnerships can we build? It's very important, even as students. My students still build partnership because I give them the flexibility. So you see my student talking to someone in the engineering, a professor in the engineering department, because he had to use equipment there to do something. Or maybe in a conference like this, a student presents something from animal science, but my student can apply it in his or her research. So eventually we connect. So being in this conference is very useful. Try to learn from what others are doing, the cool stuff that others are doing, and then take it up. It will help you because you have to be innovative. And then reimagining tomorrow is not a single act, but a collective journey. And like all things in horticulture, it begins with a seed. I'm saying horticulture because I'm a horticulturist. So a seed of innovation, a seed of collaboration, a seed of hope. We should think about all this. And thank you. So these are some publications that we've made from this presentation so far. and then so I would like to acknowledge my funding agencies too. And thank you very much.
Thank you, Prof. Let's see if there is any question, maybe one or two to the speaker. Yes, Professor Mariga.
Thank you so much, Dr. Abi. My name is Professor Mariga, as my fellow prof has put it. I'm the Dean School of Agriculture and Food Science, and to start with, I'm really grateful for the work you bring with my students, Chapita, and now Betty will be coming, and you have more there. Thank you for giving them that opportunity to be exposed to the world of research, and as a university and school, we're grateful. Mine was just but a great gratitude message to you. Thank you so much, Professor, and also for the work you're doing with our continent. I know you know some of our professors, when they go abroad and have that golden opportunity that you have, they don't give back to the community. But we hear what you're doing in Tanzania, Kenya, your country. Keep it up, Prof, and thank you so much. We really appreciate it. And Prof Masinde, thank you for bringing Professor Lodabi every conference. Thank you, Prof.
Thank you very much, and thank you for having me, and I really appreciate Dr. Masindi and the VC, because this conference, the VC called me and personally on phone to ask me to come after Dr. Masindi has contacted me. So I really appreciate, and all my collaborators and the university and others. Yeah, I feel like this is my second institution. So.
Yeah, thank you. Was there a hand up again?
Yeah. Quick question. Thank you for the presentation. Professor, in this area, we already have a lot of variation in soils. How much does your research cater for different, like in here, let's talk about like oxasols, vertisols, alpha sols. Does that have a large bearing on the results of research? And link that same question, the issue of cation exchange. Since you say we're not going to feed the world of organic agriculture, should be encouraged like a regime of composting together with other inputs. Thank you.
Yeah, so because I'm not a soil scientist specifically, I don't deal with the physical, the different physical features of the soil, like as you said, vertisols and all those kind of things. But what I do is that I get the soil and then look at its health, right? So it doesn't matter what type of soil is or what classification of soil. That's not my major concern. My major concern is if I'm giving a clay soil, for instance, how can the clay soil be enhanced to improve productivity and quality of food? How can we derive help from that soil? If I have answered your question. Is that?
Yeah, I know in Canada you don't have the same kind of variation.
Yeah, exactly. Yeah, and in Canada too, we don't have much variation because our soils are not old like in Africa. African soils are very, very old, so not have that variation or But the compost work was done in soil. I think I've forgotten I think ortisols or something like that. Yeah, that's what the soil that we use for.
We have a lot of those too.
Yes. Yeah. Yeah. So and then you mentioned the second question was
Cation exchange.
Yeah. So cation exchange is enhanced by compost. What I like about compost is that it has that buffering capacity, so it doesn't allow soils to go too far to the extremes, right? Either low pH, high pH, and by that it helps with maintaining the regime of cation exchange. Plus, you are adding nutrients anyway, so you intend to improve the CEC of the soils as well. And we took measurements of the CECs, and they were enhanced by compost. So, yeah.
There is another question. I think that will be that's going to be the last one because of time.
Thank you so much for this opportunity. My name is Betty Kare. I'm Betty that Prof is talking about. We'll be working with him, and I thank you for the opportunity. So, through the presentation, I've been amazed, and of course, I had some triggers within my mind. For example, I think there is the emphasis on the use of compost. And when even as a farmer, I was thinking to myself, I think for our farmers, we have more than enough inputs. If you consider the large quantities farmers want to apply at once to the farm. So I had that in mind, and we need to think about it, because at the back of my mind, I'm like, okay, these are our farmers have enough inputs to do more production, though they are overusing it in a small piece of land. So, and then again, from the basis of food security, or rather as a food scientist, I was one of these persons who thinks that organic farming could be a solution, maybe due to the challenge of pesticides, residues, and heavy metals. But from what Prof. Abi has said, I have to rethink about it, because also, other than, in addition to safety, we have a growing population. So I'm forced to reimagine tomorrow in regard to feeding the whole population, which is expected. Thank you for the good presentation.
Thank you. That's a fair comment.
Thank you. Please, let's give him more clap for that. Thank you, Prof. Now, Lucy, is your here with us?
Yes, I shall.
Please, as you presented others that will be coming, it is strictly 10 minutes. And if 10 reaches, you stop where you are reached. So ensure that it is 10 minutes. I will not be unfair to anybody. Once 10 reaches, that's the end. Thank you.
Good afternoon, all. My name is Lucy Gacheri, an MSc student, and I'm presenting a topic on influence of different types of organic manure on soil moisture, growth, and yield of slender leaf. Slender leaf is scientifically referred to as the Crotalaria. My supervisors are Professor Gitonga Kanata and Professor Peter Masinde. As part of introduction, I'll start by saying that slender leaf is one of the leguminous crop or plant that we have, which is known to have the ability to fix nitrogen and therefore improving the fertility of the soil. It is also known to be rich in protein, vitamins, as well as other minerals, and that is according to Muniaf et al. In 2015. When it comes to the production of slender leaf, there are certain challenges that have been faced. One of the challenges being a crop that has been underutilized as a result of limited research work as well as extension support. There is also the challenge of poor seed system as well as low input use when it comes to its production. And also we see its nutritional value has not been fully harnessed. In terms of the organic manure that I was using in this particular study, the organic manure is known to improve fertility of the soil as well as the soil health, and also it helps in the recycling of the nutrients in the soil according to Bezal et al., which is work that was done in 2024. It is also known to promote microbial activity and microbial diversity as well as long-term productivity. And because of that, it is environmentally friendly and it is also locally available, which is also used in the improvement of the moisture of the soil. In this particular experiment, or the work that I did, I used 2 different types of organic manure. That is the goat manure, the poultry manure, as well as the cattle manure that are clearly shown in those images. On the statement of the problem, soil fertility in sub-Saharan Africa and also in Kenya, it is declining. And there's a lot of use of the synthetic fertilizers that are posing environmental and economic challenges. So, sustainable soil fertility management is urgently needed. When it comes to the slender leaf, it remains an underutilized crop due to its limited research work, weak extension support, poor seed system, and the low input in farming despite its significant untapped nutritional potential. And justification, slender leaf is a nutrient-dense crop and it is well adapted to very local conditions. The crop can contribute to both nutritional security as well as the improvement of soil fertility. Organic manures, they enhance soil health and they support sustainable crop productions. Therefore, integrating the organic manure into slender leaf production offers an environmentally friendly alternative. The main objective or general objective was to evaluate the effects of different types of organic manure on soil moisture, growth, as well as the yield of slender leaf. Specific objectives, number 1 was to determine the effects of different organic manure on growth and yield of slender leaf. And the second one was to determine the effects of different organic manure on the soil moisture as well as the regulation of slender leaf. The hypothesis is there with different sources of organic manure as effect on growth and yield of slender leaf. And also the second hypothesis is that different organic manure has an effect on soil moisture and root nodulation of slender leaf. On the materials and methods, this experiment was carried out or was conducted at the Meru University of Science and Technology, that is in the demo farm or demonstration farm. It was laid down in a randomized complete block design with 5 treatments. The first treatment being cattle manure, second one goat manure, poultry manure, NPK, and then there was a control, and it was replicated 4 times. The seeds were directly planted at a spacing of 30 by 30 centimeters. On data collection, the following data was collected. 1, soil moisture, the data on soil moisture, the data on growth parameters, which includes the plant height, the number of leaves, the diameter of the stem, chlorophyll index, among others. Then there was the data on yields as well as the data on regulation. On data analysis was done using the SAS software and it was subjected to comparing of the means, that is the least significant difference to determine the statistical differences. And then the data was presented in bar graphs as well as tables. On the results and discussion, I'll start with the first one, which is on the effect on root nodulation. That is the effect of this organic manure on the nodulation of the slender leaf. So we have the data in two seasons there. A is season 1 and B is season 2. And looking across those two seasons, the nodules per plant, the nodule number per plant was significantly affected by the manure type in both seasons. The highest nodulation was recorded in NPK, that is at the 3.2, and poultry at 36.2, followed closely by goat at 34.5, then the cattle manure at 29.5, and then control at 21.7. And from this, we can see that poultry manure increased nodulation by 157% in comparison to the control. And this suggests that organic manures, they enhance nodulation via improved soil microbial activity as well as availability of nutrients. That is according to the work that was carried out by Zhao and Zubagen. On the chlorophyll index, again we have the data in two seasons, and looking at that particular result, we can also say that manure sources, they significantly affected or influenced the chlorophyll index in slender leaf in both seasons. Looking at poultry manure, it consistently produced the highest chlorophyll values, reaching to 57.4 spand and 62.0 spand in the second season. The control treatment, again, on the other side, they recorded the lowest chlorophyll values throughout the study. And higher chlorophyll content under the poultry manure indicates improved nitrogen availability, which enhanced leaf greenness, and to a great extent bringing about the photosynthetic activity to an increase. On the effects on the seed weight, again we have the data for the two seasons, the results for the two seasons there. And again, looking at season 1 and season 2, we can say there was significant, that was recorded, so organic manure application significantly increased the seed weight of the slender leaf in both seasons. The poultry manure recorded the highest seed weight, followed by NPK, goat manure, cattle manure, and control recorded the least. And this indicates the importance of nutrient supplementation. Still, we can say that higher seed weight under poultry manure could be due to its rapid nutrient release and greater availability of nitrogen, phosphorus, and potassium, which enhances the development and the filling of the seeds. And according to Ojuang, in 2022, he reported that farmyard manure significantly increases sesame seed yield to up to 148.1 kgs per hectare at 15 tons per hectare application rate. On the fresh weight, again looking at season 1 and season 2, we can see the results, whereby the fresh weight was significantly again affected by the manure that was used in both seasons, with the poultry manure again consistently producing the highest fresh biomass, and it was followed by NPK, then goat manure, and finally we had the cattle manure, while again the control recording the least fresh weight or the lowest fresh weight. The higher performance of poultry manure is linked to its rapid mineralization and high availability of nitrogen, phosphorus, and potassium, which enhances the expansion of the leaves, the chlorophyll formation, and the photosynthetic efficiency. And according to Soliman, he reported that poultry manure applied at 4 tons per hectare increased the amaranth fresh weight to up to 26.99 kgs.
I'm sorry, time is up.
I'm done already. We have the references. Thank you.
When I set my timer, when it's ready, you are timed back. Thank you so much. I will not ask you to ask questions now. Let's see, ask questions after maybe 3 presentations. So please, hold your houses. Write down the question, hold it. After 3 presenters, you can ask your questions. Thank you. So you are the next. Just want to be sure that it is Jane. You are Jane? Good.
Good afternoon. As the presentation stands on, I want to introduce myself. My name is Anne from Google. I'm a PhD student in sanitation and waste management. And today I am presenting part of my thesis work, and the topic So I want to appreciate my supervisors and my co-authors, Dr. Joy Yongo and Dr. Stephen Karanja, who is seated right there, and Dr. James Keremi, who has worked with me throughout, who has been with me throughout this journey. And today we want to talk about the black soldier fry, enhancing nutrient composition for diets for rearing the Nile tilapia fish. Enhancing nutrient composition for diets for the Nile tilapia fish production. So as a simple introduction, we are saying the world's population is increasing, Kenyan population is increasing. Without having adverse effects to the environment, because especially when we want to produce food, we go cutting down our forests. So other ways of producing food for the high population has had adverse effects on our climate. So we want to look for methods that we can still feed out the high growing population and at the same time manage the high volumes of wastes that are being produced. So as we provide food to the ever-growing population, we also want to provide quality food. So fish has been identified as a good source of micronutrients, and hence we need to look at a sustainable way of producing fish. One of the main ingredients that is used in formulating fish feed is the fish meal. However, due to the changes in the climate and decreasing resources in the sources, then we are having challenges and overall increasing the cost of production. And hence, this study really looks at a way that we can effectively or sustainably replace the decreasing fish meal portion that is used in fish feed production and replace it with our black soldier fly, which studies have shown that it can be able to provide necessary nutrients. So moving ahead, this study was carried out here in MAST, and specifically the Sanitation Research Institute, and the black soldier fly was reared, and the environmental conditions were indicated there, studied earlier by other researchers within the region. The substrate that were used to rear the black soldier fly is our organic waste combined with the fecal waste. So it was processed after rearing, it was dried, it was defatted, and feed was formulated. So during the formulation of the food, 3 isonitrogenous diets were prepared. Diet 1, which had aimed at still utilizing the 100%. So the portion of the protein that we are replacing is not the whole protein, but the portion that is commercially given by the fish meal. So if you look at that table, in diet 1, then we are still retaining the whole portion of the fish meal. In diet 2, we are replacing it 50 by 50%, that is the 11% there, and the diet 3 has whole substitution of the fish meal portion with the BSFL. So it was prepared, and we still have other sources of the proteins, like from the soybean, the sunflower, but this study aimed at really replacing the fish meal portion. So that's a pictorial of preparing the diets, make it into pellets so that when you are feeding it to the fish, then it does not sink to the bottom and interfering with the water quality conditions, and it is able to be taken by the fish when it is still in the suspended form. So we are saying that wanted to look at the nutritional components of the ingredients as well as the diets that have been prepared, and the standard protocols were used, and data again was analyzed using ANOVA, and very significant difference. So these are the results. So the study started by analyzing the proximate analysis for the ingredients. So we have the ingredients, the soybean, the fish meal, all the way to the black soldier fly larva meal, the BSFL column there. So looking at those results, notably, there are significant differences, and of key to note is the close relationship in the crude protein that is found in the fish meal versus what is found in the BSF meal. So comparing it to these other sources like the plant sources, soybean, then the BSFL still appear superior in comparison to other sources. The limitation that we may give to the BSFL is limited ash, and moving ahead, an analysis of the diets that we have said were prepared by 0 replacement, 50% replacement, and 100% replacement. Again, values varied significantly except for the dry matter, but there are also high values of dry matters indicating the storage as well as the nutrient density that can be in those feeds. Again, notably, is the food protein value. So their value, they varied across the 3 diets, but again, diet 2 emerged superior with the food protein level. We are really interested in the food protein because it helps in the overall growth and will support the growth of the fish. When you go to the fiber, the good fiber value is higher in diet 3. Good fiber, and specifically the acid-guided fiber, which is higher there in diet 3, the value 13.39, talks of the indigestible part of the carbohydrates, the cheating, and hence limiting the use of the diet 3. When you go to the fatty acids analysis, again, diet 1 still emerged superior in the saturated fatty acids and the monounsaturated fatty acids, but when we go to diet 2, it emerged superior in the polyunsaturated fatty acids. So we look at again, if you look at the diet 2 and diet 3, they are still rich in the lauric acid, that is the C14. They are still rich in the palmitic acid, the C16, and notably its superiority in the C18, the stearic acid. Stearic acid, again, can further be synthesized into the long-chain polyunsaturated fatty acids, and also reducing the aspect of the bad cholesterol that could be found there. We go to the C18, what we are calling the malic acid, and yeah, it is still there, and notably I talked about the C18, which is the linoleic acid. Linoleic acid can further be synthesized, desaturated, elongated to give us the long-chain polyunsaturated fatty acids like the EPA and the DNA, in simple terms, our omega-6 and the omega-3. So we are saying that, in conclusion, that diet 2 still became superior in the food proteins and in polyunsaturated fatty acids, and these 50% help us.
Thank you so much. Let's appreciate her, please. Chen Katwiri, you should be here on the stage.
Thank you, Chair. All protocols observed. Good afternoon. My name is Jane Gatweri. I'm a PhD student in the Mathematics Department, the School of Pure and Applied Sciences. I'm taking a PhD in Mathematics, and today I'm here to present our work that we've been working on with Professor Eustace Mwenda and Dr. Stephen Karanja on the finite volume-based numerical solution framework for analyzing confined buoyancy-driven turbulent flow field, as he organizes that. So I'm a PhD student. The finite volume method is one of the numerical methods that are used to solve fluid flow problems among the many that exist. And in our research, we used the finite volume method because the finite volume method has been proven to be more robust to other discretization methods. So, as I said, these are Thank you very much. So this is our work, finite volume method-based numerical solution frameworks for analyzing confined buoyancy-driven turbulent flow field. I am the presenter, Jane Gatweri, and I've been supervised by Dr. Stephen Karanja and Professor Eustace Mwenda. As part of the introduction, you're saying that buoyancy type of flows. These type of flows can be modeled using various methods. One of the methods is experimentation. The other method is use of analytical methods. Another method is the numerical approaches. In our case, we made use of the numerical approaches. There are three main: the finite element method, the finite volume method, and the finite differences. We chose the finite volume method because of its geometrical flexibility, because it can be used on any type of geometry, and its robustness in conserving the conservation laws that govern fluid flows. The finite volume method does not work in isolation. It requires coupling with other numerical components. These numerical components are, part of them are the interpolation schemes, the pressure velocity coupling schemes, and the turbulence models. The accuracy of the solution that is finally obtained is largely determined by the components which are coupled with the finite volume method to generate a numerical solution, and therefore it is necessary that we look at these components. We had 5 study objectives. One was to determine a numerical solution framework for analyzing velocity profiles. We were also looking for a framework for temperature, a framework for turbulent kinetic energy, and a framework for ND viscosity, and finally to come up with a solution framework for analyzing buoyancy-driven turbulent flows. In the related studies, among the many that we had, I've highlighted these two. We have Berger 2021, who did a study on the simple, simplex, and simpler. These are called pressure velocity coupling schemes, and this study concentrated on only the pressure velocity coupling schemes, which is one of the components of the finite volume method. Then we have Komen in 2022, who concentrated on other components known as the interpolation schemes. Komen was dealing with the upper differential scheme as well as the central differential scheme, coupled with the finite volume method. So looking at these two, among the very many, we are finding that research has been done on the finite volume method, but the researchers have concentrated on a particular component instead of looking at all the components as an integrated system. And now this led to the system of our problem that despite the significant advancements in computational fluid dynamics, there is no explicit solution framework for analyzing buoyancy-driven turbulent flows, because the previous researchers have taken the numerical components like interpolation schemes, pressure velocity coupling schemes in isolation rather than as integrated system, and therefore there is no unified consensus on the optimal combination of numerical components to obtain a numerical results that mimic a realistic flow field. Therefore, there was need for a systematic evaluation of numerical components that can be coupled with the finite volume method to profile a flow variable in buoyancy-driven turbulent flow fields. In our methodology, we started by defining the general laws of physics that govern fluid flows, that is the equation of mass, momentum, and energy. A lot of work was done here on these equations to get the equations that govern a general turbulent flow field using a process that we call Reynolds decomposition and averaging that gave us another set of equations. These turbulence equations were subjected to a process that we call discretization to give us, to convert the continuous equations into algebraic equations that could be solved. And finally, we used the interpolation schemes which were first order upwind, power law, and quick to obtain spatial interpolation, as well as PISO and simplex, which are pressure velocity coupling schemes. So if you look at our methodology, we do not deal with only one component like the previous researchers. We are having a unified combination of components. When it comes to pressure velocity components, we didn't look at one, we had two. When we came to the interpolation schemes, we didn't deal with one, we compared three. And therefore, and then the results that we got, which are numerical results, we again validated them against benchmark results of Ampofo and Karayanis 2003, specifically chosen because Ampofo and Karayanis had done a similar study, but they've done it experimentally. These are the governing laws. I've not brought all the equations, I've only given the general. These are the first one, equation number one is the general mass conservation equation, momentum, then energy. These equations underwent a series of processes until we finally got the final set of turbulence equations, which are presented here. In the interest of time, we may not explain all of them. Those equations were solved using a software known as Fluent software. In the Fluent software, we had the advantage of having to produce several sets of results again. We had results in terms of contours. The contours were explaining the flow, the general flow field in the flow domain, and we were able to get the regions of which were the regions of high velocity, which were the regions of high temperature distribution, and then finally now we got our second set of results which are presented here for purposes of validation with the results of Ampofo and Karayanis. So I've presented only the validated results of Ampofo and Karayanis. As I said, we had used three discretization interpolation schemes. One is the power law scheme, which is given by color green. We have the upwind differencing, which is given by color blue, and the quick scheme, which is color purple. And from these results, which are validated against the black, which is experimental, we can clearly see that the quick interpolation scheme gave us the results which closely matched experimental results. This happened for all the flow variables. The results you can see are for velocity distribution in the cavity. We had results for temperature distribution in the cavity, which is a square cavity. Again, the quick scheme gave us the results which closely matched the experimental data. Also the kinetic energy and also the eddy viscosity distribution in the cavity. And from the results that we got, we came up now with the following conclusion, that the solution framework for all the examined four variables, that is temperature distribution in the cavity, velocity distribution in the cavity, turbulent eddy viscosity distribution, and the turbulent kinetic energy distribution, the framework is as follows. We recommend the use of the finite volume discretization scheme, and we recommend the use of the SSTK omega turbulence model, which is the validated model that we used. We recommend the pressure velocity coupling scheme as PISO. This because we validated PISO against simplex. We chose simplex and PISO because these are the two most commonly used pressure velocity coupling schemes, so we decided to test these two, and PISO emerged superior to simplex, and therefore we recommend the use of PISO. PISO means pressure implicit with the splitting of operators scheme. For spatial interpolation of the flow variables between nodal points, we tested the power law scheme, the upwind differencing scheme, and the quick scheme. Quick means quadratic upwind interpolation scheme for kinematics. And now we recommend the quick scheme for spatial interpolation. This combination of numerical components constituted an optimal framework for analyzing confined buoyancy-driven flow fields. Based on the findings of our research, therefore, we recommend the use of this solution framework in the following areas. We are reimagining tomorrow, And we were encouraged this morning that it should be an interdisciplinary approach. So in mathematics, we don't work in isolation. We work with computer scientists and many other engineers. So we are recommending this usage.
Thank you so much.
Thank you, sir.
To appreciate her. She's a timekeeper, right? I think she's done well in keeping time. She was already concluding anyway. So she's setting the pace for us as a PhD student. So those coming, you can see, you can manage in less than 10 minutes. There is someone coming next, and I'm not seeing her. McKenna Lena, are you here? Because we wanted to do away with the morning session before we break. Just one more. Where is Lena McKenna? Any information? Because we will not give you another opportunity. And let's. You should even not present. No wonder then the student has not come. Because the supervisor is not even aware. You don't allow a student work without the name of a supervisor. I've never seen it. So please let's practice things the way they're supposed to be done. We can replace with who? Because we just want one more. We go for lunch. And then when we come, we'll be asking questions. Yes. Sarah? Are you here? Yes, please come. Kindly.
So, good afternoon. My name is Sarah Wanja Riongo. I'm a master's student in the Department of Biological Science, Melbourne University of Science and Technology. I'm going to present part of my research work, elevation and restoration shape the distribution and intensity of Lantana Camara inversion in Waimenti Forest Kenya. This study is under the sub-theme International Partnership for Achieving the SDGs, and I was supervised by Professor John Moria and Dr. Peter Kebenji. Forests are very vital, as you all know. They are very important for biodiversity, climate regulation, carbon sequestration, and even improvement of livelihood. And then biological invasion has emerged as the critical driver of ecological changes, causing biodiversity loss and ecosystem degradation. Now, in this one particular, Lantana Camara has been rated as the worst, one of the worst invasive weed or species. And in Kenya, it has invaded many ecosystem areas, the forest, the protected areas, and even the wetlands. And what Lantana Camara does, it suppresses native regeneration. It affects the forest richness, causing overall degradation. Now, two factors that are likely to affect the invasion dynamics are elevation and the intervention, the restoration intervention. Elevation affects the environmental conditions, such as the temperature, the moisture, which potentially influence the invasion or the spread. On the other hand, restoration intervention influence, the restoration may control the invasion by mechanical removal and even kind of recover, the native kind of recovery. Now, these interventions, their effectiveness have not really been understood, and therefore, limited research has examined how the elevation and the intervention, the restoration interventions gently influence the Regional Camara invasion, especially in Kenya mountain forest. As it continues spreading within the forest ecosystem, it poses significant threat to biodiversity and restoration efforts. The previous studies have looked and documented the Regional Camara impact, ecological impact, and its distribution. It has looked at how the species occur, the habitat suitability, but limited understanding exists on how the inversion varies along the gradient elevation, and also whether the restoration efforts are effective in reducing the invasion. This study was guided by 3 specific objectives. First, to assess the spatial distribution and invasion intensity across the forest. And 2, to evaluate invasion variation along the elevation gradient. Objective number 3 was to assess the influence of restoration status on invasion. Therefore, this study was conducted in Waineti Forest, which covers roughly 2,462 hectares. It is one of the part of Mount Kenya Forest, which is a biodiversity hotspot, and it is experiencing this threat. A stratified sampling was adopted, and therefore the forest was stratified into 3 zones, spanning between 1,200 meters above the sea level to 1,500 meters above sea level. Therefore, we had 3 study zones: the lower zone, the mid zone, and the upper zone, the higher zone. Across all those 3 zones, we established 3 belt transects to represent the management status, the ongoing restoration, the unrestored sites, and the sites which were previously restored. We established 40 samples systematically across the elevation. And the Regional Camara now was estimated visually using the Brown-Branke scale. On the overall distribution and pattern, the distribution was heterogeneous. It was wide. From figure 2, we can see there are plots that recorded close to 100% invasion, and there are others that recorded almost 0. The mean coverage was again high. It was about 64.08%, with a wide variability of 35.68%. The range was between 2 to 99%. On category C, according to the Brown-Branke scale, where we have the invasion in depth, majority of the plots fell under the high intensity, showing that the forest is heavily saturated with Regional Camara. Now, this result shows an overall bimodal invasion pattern, whereby we see some plots are heavily saturated with Regional Camara, while others are not. So that's the pattern we observe there. The loan was, we assessed the elevation gradient trend, and the elevation intensity differed significantly across the three elevation zones. The high elevation had the highest lantana cover at 82.5%. Low elevation had around 56, and the mid elevation showed the lowest. Now this one is a deviation from the mid elevation peak theory. It was confirmed by box plots, which showed that the high elevation had the highest lantana cover. In fact, we observed a uniform saturation pattern. The middle elevation showed the lowest, as you can see, and it showed biotic resistant. It is completely resistant, and this could be attributed to the restoration efforts and canopy corrosion. However, the lower zone showed patchy pattern, and this one could be attributed to early stages of invasion. On the restoration efficacy, this is where we have the restoration interventions. The ongoing restoration site maintained near to zero lantana cover. When we conducted the Fuscowalis test, it showed that restoration structures had the strongest influence on invasion pattern, with a large magnitude effect of 0.41, telling us that intervention explained around 41% of the lantana cover.
Thank you so much for that presentation. I want us to appreciate her, please. Now, I want to make some announcements. I know we have not asked questions for about 4 presenters already done. I would like you to write your questions down. Maybe it will save us time when we are going for lunch and we come back to respond to those questions. You can have your questions written down in the past over to us, and then we can read them also out. And maybe sometimes I find getting a microphone to everywhere where he or she seated is becoming more of a problem. That can be of much help. I am supposed to make another announcement, but because the registrar is with us, I think you will be best blessed to make this announcement. Registrar, I know in the afternoon session we are supposed to have our VC together, some visitor. I think you will be the best person to make this announcement to us so that we can adhere to the request. Thank you.
Thank you. Let us give Dr. Professor Wamaro a clap. Thank you for reading us very well. We are truly grateful to all the presenters. We are really grateful to our students. Now, in the interest of time, exactly at 2, I know we are actually about 24 minutes away. We have presenters from Italy, and we have adjusted the program slightly because of issues that we all know. So we are supposed to be here exactly at 2. Exactly at 2. In fact, we are supposed to fill all the seats so that we can actually present to a presenter from Italy on precision agriculture. Precision agriculture. I hope Professor Doita have got it right. It is precision agriculture. So please let us be here. I believe you are directed on how we are going to take the meals. The VVIP, I think we are still using the tent. And the rest we actually, you can guide us a little bit. Yeah, just guide us so that immediately we leave here, we take our lunch to exactly we be seated.
Thank you, Registrar. So we will still take our meals the same way we took our snacks, the same venues. So the VVIPs, the guests, the rapporteurs, session chairs, deans and directors will still have our lunch at the tent behind here. Then the presenters will have our lunch at ECA 09. The audience and staff will have the lunch at ECA 05 and the ICT and security at ECA 07. I hope that is clear. It's not clear, it's not the same. I'm being corrected, I'm being reimagined. Okay, there is some slight change that the presenters should take their lunch at the cafeteria. That's all. The rest is the same. So I think from here, Registrar, we go for lunch and then we come back at 2. We can go for lunch. So all the presenters will go to the cafeteria. The VVIP guests, the deans, directors, rapporteurs, session chairs will go to the tent. The faster we do it, the better, so that we are here exactly at 2.
what you have taught them in class, something very, very good. Through them, we have also taken some of our students to some of the best mechanized farms in Naivasha at their own cost. So this is a collaboration which is really picking up. I don't know where Dr. Eric Moria is. Eric Moria has been like a consultant in that farm, and we are very happy of the work that we have continued to do. And every year, from last year, we always have a spokesperson from the group coming. And even this afternoon, we have Mateo, who runs a big farm, one of the biggest farms in Europe. And we are very happy that you always, when we call you, you come. I'm very happy. But I don't want to introduce them. I would like to call my friend, who is the vice president of the Rotarian Club, my friend Magda. Please, you can make a few remarks, introduce them, and we call Mateo to come. Thank you very much. You can clap for her.
Good afternoon, everybody. And first of all, I would like to express my gratitude to the Vice Chancellor, Professor Romano Sodiambo, for his kind invitation. My name is Magda Pagetti. I represent the Rotary Club of Monza Brianza. It's in Italy, near to Milan. And it's a great honor for me to participate to the conference, Mustit conference for the second year. And our club, Rotary Club, supports the San Francesco Association, which has been active in Kenya for 25 years. Thanks to the high dedication of its president, Giancarlo Magni, the association founded the the St. Francis Village here in Enchiro. Currently, we are developing a farm and an agricultural school in Chiamuri. Our goal is to implement precision agriculture technology, and with the support of Matteo Antonello, that is the general manager of Spectra Agri in Italy, that will share more details about this important project shortly. Now I would like to introduce my colleagues and friends that are with me today. First of all, Giancarlo Magni, se ti puoi alzare, Giancarlo Magni, President of San Francesco Association. The second is Matteo Antonello, he's a special speaker. He's a general manager of Spectra Agri and is also a Rotarian. Francesco Teruzzi, he's a Rotarian and is a member of the club. He manages the financial and administration aspects for the Chiamuri farm. Ilaria Teruzzi is a Francesco daughter, is a member of the association and is involved in the project. Claudia Crespi, she is closely with Francesco on the project for the administration aspects. Giuseppino Scida, members of the San Francesco Association, and not the last, but the first person is Luisa Maina, manager in charge of the farm operation in Chiamuri. Thanks again for your attention and for wonderful opportunity. I wish you all a highly productive conference that will inspire the university and also the entire community. Now I will give the floor to Matteo Antonello from Spectra Agri. Thank you very much.
Thank you. Matteo, you can just stand. I really wanted to recognize Father Rewa. Father Rewa, you remember, introduced me to your Italian friends, and now I've become part and parcel of it. I'm very happy, and thank you for the great work that you are doing the whole world to transform, impact, and change communities. So we really appreciate you. Thank you very much.
Thanks, Professor Romanus, and thanks to Mero University to welcome us here again this year. It is a pleasure and an honor for me to be again one more time here. Before starting, I would like to thank the President Giancarlo Magni and all the Rotary Club of Monza Piazza, together with all the friends together. It's my second year in a row that I'm coming here, and my wish is to come here every year, maybe more time a year. Okay, so I will try to make the best presentation as I can, and trying to underline what the technology is doing all around the world. So my job is managing a company that is truly committed to agriculture, but precision agriculture. Something that in a continent like Africa could look very far away, but in reality it is not. Agriculture is something that is very important. Actually, is fundamental to the life. The more we are able to produce, the more we are able to feed the people and the persons, together with the animals and so on. So agriculture is life. The company where I work has a commitment to try to improve the operation within the agricultural sector, putting technology within the machinery, but also in the people that are using technology. There is no technology without people. So the first thing that I would like to underline before starting is again that without people, we don't go nowhere. So let me start, because last year I was here talking for a few hours actually about precision agriculture, and I want to recall just a little concept for the ones that were not here. But I'm glad to see that many people came back to see again this presentation. So, what is agriculture? Precision agriculture is something that again you can support the development of agriculture throughout technology. It's a passage through the traditional agriculture. So what we are used to do now, and what we could do, or what other people in the world are really doing, is a way to use better the soil, better the inputs, better whatever we can do to improve the yield, and again, make more food available. So precision agriculture, under the real aspect, what is really technology agriculture, is the addition of technology, of knowledge, of sensors, of robotics to the everyday operations in order to improve what you are really doing. Why and how? Knowing first what you are doing, knowing in which field you are working, what is in that field, what the kind of soil it is, and try out the best from it. And this is something that you cannot do just watching the crop and hoping that the weather will be good. It's something that you have to really analyze and discover and support. Precision agriculture has this mandate to support the development of agriculture using the technology that is available. So what are the main goals of precision agriculture? Of course, maximize the yield. Means to have from the same amount of land, more product. More product means more food. But without wasting soil, without utilizing too much fertilizer, without using what is not needed from the crop. Maximize yield thanks to what is the crop really needing for. And then, of course, reduce the input waste. How many of you think that agriculture now is really biological or organic? I personally don't believe that it's really biological organic. But I do know that there are a lot of fertilizers, pesticides that have been used and are used without any real reason. Precision agriculture helps you to reduce the amount of this kind of things. And then, of course, because at the end of the day, every little farm is a company, improve profitability. A farmer that can get something from his production is a farmer that is happy, can pay food for his field, and then can send his sons and kids to the school and so on. And then, another very important aspect, to protect the environment. Utilization of less input, of course, create less input in the soil, in the air, and whatever we do. And, very important, I took it as the last point, empower farmers. I tell you that a farmer that is using precision farming is a stronger one, because it's a farmer that understood how technology and agriculture can work together and utilize his knowledge to better and improve day by day. So it's an instrument, it's an implement to really empower the people that are working in the farm. So this is a brief resume of what I did and what I said last year. What I want you to show you this year is something different. Because, guys, I am the first that is going to a meeting and telling about how cool is precision farming. But what is the reality? And this time I wanted to take you an example of a farm in Italy. I'm not talking about a huge farm, I'm talking about a standard farm. And I want to take the case, this farm, to you, just to put your attention that it is absolutely possible. It's a company that is called Parma, that is the name of the farmer, technology farm. We call it because the utilization of technology is really at the top in Italy. But before, I want to make a question for you. Can a family farm increase corn production by more than 20% without buying more land? So it's possible that a family farm, produce more without buying more land to grow the corn. No answer? Let's say yes.
Yes.
Yes. Yes. Good. Very good. The answer is correct. So this is exactly the story of this. This is a story of Parma Technology Farm. So as you know, this is Italy, and this farm is located in the north part of Italy. So let's say the most agricultural area. Why? Because there is a lot of water, and there is historically a very developed agriculture. But also, very important and very easy, is a very, very big flat land. So, this company, this farm, was born 4 generations ago. So, I'm not talking about a company that, a farm that started 5 years ago. It's not a new one. It's a company that started after the Second World War, when the agriculture was, yes, still with cows and plows, pulled by cows and bulls. So they started 4 generations ago, and now the grandsons are taking the lead. So it's founded, in 1960. So and they do two big things. They have the livestock. They have 220 cows for beef, so for human consumption, and then of course to feed the cows, they have the production of corn for the livestock feeding. I put a smile because the blue shape is the region of Lombardy where this farm is located. So implementing new technology into this farm. But where they started from? So as I said, many years ago, the agriculture in Italy was a standard, as we call it standard. But what they were really missing compared to today was, of course, a limited data from the field. What do I mean? They were, of course, planting, harvesting, crop caring, but they were doing it as of today, doing it day by day and day by day. So they were not recording whatever they were doing. So all the fields were managed at the same the same system. No difference between a field that was, I don't know, close to a river or another one that was close to a hill. Of course, this was also translated into profitability. They had to feed the cows, because the cows are, let me say, not like here, a bit bigger in Europe. They can weigh 300 kilos and they eat a lot. So they had also to find a way to feed more and more the cows. And all the decisions were taken by expertise. So the grandfathers were saying, okay, this year do this in the field and do that. So there was no recording. There was just the experience that was guiding it. But they said, okay, is that possible that we can produce more with the same land using these habits? No, it was impossible. So there was a need for a change. From traditional farming, they started to collect data. They started to take with the data better decisions, and at the end of the day, you will see now this presentation, they get better yield. So the last generation, so 25 years old young farmers, said, okay, we can, if you want to increase our profitability and our yield, we need to make a game-changing decision. We go into precision farming technologies. And so, what I'm going to show you in the next slides is what they did in the last 5 years again, not in the last 26 years. In the last 5 years, they started, first of all, collecting information, and then they applied this information to the everyday and seasonal operation. The first thing they did was this. They started to transform their land in a digital land. So they started to really scout the fields and the operation in order to understand what was in the field. There was water, there was nitrogen, what was in the field, and then put some operation accordingly. So they started to monitorize the fields. They started to use a GPS picture to understand where maybe there was water standing somewhere, or maybe taking picture about the leaves from the satellite eyes to see what was the greenness of the plantation. And this started to make them think about what to do and try to find out some way to use as better as they can this information. The second step they did, and this was 2020, so at the beginning they were just scouting. They started, okay, we got all this information, we have, you see here, these are pictures of the field taken from the satellite eyes. The first picture in the bottom high is the percentage of water standing into the soil. The second picture is about a crop and the greenness of the leaves of the crop plant. From this operation, they understood if there was some diversity within the field, if there were some operation to do. The second step they did was to automation on the machine. So they started to understand that the tractors that the operators were driving every day could be improved by a few operations. So instead of driving the tractors all day long, all the season long, back and forth, going back right and left, and maybe sometimes overlapping when they were planting, or maybe they were during crop caring, overlapping when they were spreading some pesticide or fertilizer, try to automate it and use the guidance for tractors, so an automatic guidance system installed in tractor, to make it guide autonomously into the field, in a way that the tractor was not overlapping all the other swaths before. And so, you know, this was the first step. And this is important. It was an important step for the farm, because if before all the fields were like a kind of you know, not very homogeneous, and there was a lot of overlapping, and you can easily see when there are a few rows that are overlapping. From this, they started to have very, very perfect fields and no overlapping. This means that they don't have to use more inputs to overlap one more time and spread more. You see the picture here. You see So this is the tractor. So we are not talking about big tractors. This is not a big tractor. This is a 100 horsepower tractor. You can find easily here a 100 horsepower tractor. And behind there is a corn planter. In that tractor is installed a guidance system. The guidance system works throughout these displays. You can easily see that there is a display. There is a tractor, a red tractor, and you see some straight line, right? Do you see them? So these are the trajectory that the tractor is following very precisely in order to not go where is a real fast. So after the first year on the tractor, they installed this instrument that was guiding the tractor and make it possible. So, and what was the benefit? So this is exactly a picture from inside the cab of the tractor. You see that there is the steering wheel, and very important, there is nobody that is driving the tractor, because the tractor is driven by the displays and an antenna under the roof. So the operator is just there, watching the tractor to work. But what is the difference? The perfect job. You see that the display is showing you that the tractor is doing an operation, and is also painting what is done, what is doing, in order when the tractors turn not to go over. This kind of very, very, very little improvement increased the yield of 5%. Just not doing nothing. We didn't add any fertilizer, and we just drive straight. And of course, as soon as you, then after you're planting, what you have to do? You have to try to put some money into your pocket, and then you have to harvest. And so, you see this is the cornfield. So on the right you see the cornfield that is perfectly planted. You see, you can easily see how the plants are perfectly planted, perfectly straight. And another important thing, you see that the plants are at the same distance between. This is called singulation. The more singulated are your seeds, the best crop you can get. They harvested, and you see this map. This is a map that is recording where the field has produced less or more. And it's easy to see, understand that when there is a green point, means that the production has been high. When there is a red point, means that production is still low. And this is exactly the key of regional farming. So the map, the yield map, is the starting point. Once you get the regional map, the yield map, you know where your field produces less or more, in order for the next year to go on the red spots and manage them differently. Because the question here is why in the red spot we produce less than in the center of the field. Now, I believe here a lot of agronomists are here and can easily answer to my question. The answer of the question is that we need to make something for that part of the field. If you need to spray, level, do whatever, we must do in that part of the field. We don't need to spray more where there is the green dots. And so they started to record year by year the yield. And every year the yield has increased. This is important. There is an important phrase here. You cannot improve what you cannot measure. If you are not really taking care about what you do, how can you really make it better? And this is for agriculture, but this is for everything, I believe. So from 20 to 22, there was this improvement. Then they started just to guide. Let's say they started to drive better the tractor and to take some data out of the crop. A year after, they decided to make another little small improvement, but it was about the planter. So it was not about the tractor, it was about the planter, so the machine that is putting the seed under the soil. And they added these little things behind the planter. So this is agronomy, this is not technology, this is agronomy. This instrument, that things over there, the black things over there, is just a system that closes back the little furrow where the seed is planted. And this is exactly what I mean. This is a traditional planter. you see, if you put a seed in the ground, and this seed is not totally covered by the soil, let's say that she will not emerge as you want, will not get the best of the emergency. But if you put the seed in a situation when all the soil, the moisture, the nutrients are just there, this will be hopefully increase the emergency. You know that the emergency, so the first let's say the first step of the crop is the most important. If you lose the emergency, it's easy to say that you don't get any plant later, no? So you see on the right, you see all this little flag, is a way to record if the emergency was good or not. So when happened the emergency? 1 or 2 days after the planting. The goal of this test was to prove that all the little plants were emerging in the same day. And what happened? They noticed that if before they were not using this kind of implement that was not surrounding the seed by the soil, they increased the emergency of 87%. Means that the 16% of more plants came out than the year after, without doing any spraying or increase the amount of land. And this is the result. I mean, this picture maybe you remember from last year. This is a 60 days corn tall, and this is the owner of the farm. As you see, it's not an old man. He's younger than me. He's way much younger than me. He's a 32 years old man that is running this company. As long as they did this, they understood that the emergency, after an increasing of 16%, really was making a difference. So they started to fertilize the seed right after the seeding. So they were just putting liquid fertilizer during the planting in order to have to the little seed all the fertilizer that he needs for the first 2 days of his life. And this helped even a lot to increase 18% more. So I'm giving you a lot of number, percentage. I'm not here to tell you this, that this will work, but I'm telling you, using the data, using the technology, surely impact your production. So, and there we go, the harvesting. So this is an harvesting machine, an old class machine that is harvesting the crop. As you see, the crop is very dry, so they are using this for grain. Okay. But as you can easily see, the total increase of the grain was plus 22%. The total increase of the silo mix was plus 17%. So a few little steps can really improve how all the farms can work. Now, going to the end of my presentation, and thanks for listening. What is the key lesson of this few slides that I'm reporting to you? So, as I said, if you don't measure, you don't know. You will never know how to increase your yield, your operation, if you don't measure what you do. Precision farming means that all the decision has been based on data. If not, you are just losing time. And this is very important, let me tell you, in this environment. We are not talking about huge steps, a big investment. Every year, a little step can make the difference. So, this is very important. I'm learning here every day, and every day that I'm here in Africa. The technology is successful only when it's combined with agronomy. So, we don't have to think that, okay, we have technology, we are done. No. I will give you an example. The company where I work for, there are no employees that are not graduating in agronomy. So every one of them is graduating in agronomy, and because an agronomist can translate the information into operation. We have no salesman. We have agronomy that works for the technology. Now, just because I'm here, I want really to give you these few, let's say, key points to take with you. So, as I said, this is my second year that I'm visiting you, and thank you again for guesting me. But the most important reason is because I'm trying to help me and my company, the Kiamuri Farm here in Kenya. So, Kiamuri Farm is a farm that has been created 2 years ago by Mr. Gianfranco, and is, I would say, a very, very big opportunity for all the people that are around here. Why? Because in this farm, step by step, little step by little step, every day this is an improvement. So, you see here two important pictures. That means that things are already happening. Here you see, this is a satellite images of Kenya, and this is Kiamuri Farm. The orange boundaries that you see are the fields that are already being worked in the farm. What we have done, we just used the expertise of Louise, that is the managing director of the farm. And Louise, could you please stand up? So Louise is a woman that is managing director of the farm, and thank you for doing this. Helped me to put on the GPS map the boundaries of the farm. Why this? Because, as I said before, if we don't do where to go, what to do, what is the amount of land that we have to work, that is the starting point, we will never do any improvement. So we started to map all the fields. And now you can see that we have 6 fields with tomatoes, mango, corn, peanuts, and so on. And we know where they are, we know how big they are, and we can manage the operation for the next years. And the second and most important thing is thanks to a big investment from the association, roughly about 6 million of shillings, right? We got a tractor. We got a tractor that we will equip with an automatic steering system, and you will be all invited to see how it works. So we are going to install a automatic guidance system that will make this tractor drive without the operator. So, what is my message, if I can give a message for Kenya? So, precision agriculture is not about a large farm. Because I remember clearly last year, everyone was asking me, hey, you need a big farm. No. I underline one more time, you don't need a big farm. You need a big motivation to make it. Okay? It's about understanding variability, collecting data, and making better decisions. Every farm can start a digital transformation, but again, one step at a time, one little step at a time. My final takeaways. So, last year I was talking with you about precision farming. This year I took an example of an Italian farm, and I started to already talk about a Kenyan farm. Next year I would love to be invited again one more time, but to talk about this farm. This land, how can we took technology here? So, please, let's utilize this association as much as you can to get the best out of what you're doing every day. Thank you very much, and I'm at your disposal. If you have any questions.
Thank you so much. We would like to, if members who have any question to the presentation, yes, Professor Marika.
Thank you so much. Hi, Mateo. Thank you for the very good presentation, and I hope our students are here to learn more about what they learn in class precision agriculture. Mine is to thank our Italian family for the opportunity you give us as a university, and specifically the School of Agriculture and Food Science, whereby we teach precision agriculture. Before, our students were just imagining what these machines are, what precision agriculture is, but with the opportunity you gave us with Kiamuri Farm, thank you to our VC, Professor Romano Subiambo, for connecting us with the Italian family And thank you for that opportunity. Our students now visit a farm, they learn, and we hope that this technology which is learned from there will be cascaded down to the farms, and production will increase as Mateo has rightly put it. We expect to change the way we do agriculture, and thanks to you, our students have a positive feedback from the experience they get, and I'm sure they'll be practicing that in their respective areas of origin. We really appreciate as a school and also as a university for that opportunity. Thank you so much. And keep on coming back as many times as possible. Thank you.
I have to thank you for these words. I really appreciate it. We all appreciate it. So thank you very much. I mean, this is the minimum that we can do. I mean, this is not about showing what we do. And so we know that we are guests here. And what we can do is just to give you at least what is happening in the rest of the world. Okay? But let's try to do this. Let's try to make it happen here. It is possible, step by step. I tell you that I see hundreds of farms in Italy every day, in Europe and in North America every year. But you know, the improvements that you can do here are in percentage much bigger than you can do in the other countries. Because if we can get 10% more of production for next year, here will be a huge. In Europe, if you increase 10%, actually it's kind of impossible. But it's not that big. Here, to increase 10% from what you're producing now is a lot. So let's try to do it.
Another question?
Thank you for the presentation. This is the second time I'm listening to you, and I always enjoy listening to this demonstration of technology, precision agriculture. One of the pictures you've shown us there is of a young man. Oh, is it a young man or a young lady running?
I mean, this, the one on the left is a young lady, is a woman managing a company. Is her.
Okay. Yeah, at least you talked about a young person running and a precision agriculture company. And that just reminded me that in our country and in general, I think in Africa, we have a declining interest among young people in terms of agriculture. A lot of young people are not really very keen to pick up agriculture because of the view of agriculture as dirty and not very interesting for the young people. I wanted you to just comment a little bit about how to motivate young people into agriculture. Does it pay? How do you just motivate young people? Could you say a little bit more about that? Thank you.
So, thank you for this question, and thanks for coming back also this year. And this is a very good question, actually, because I tell you, it's not just about here. It's a worldwide issue. We were discussing before at lunch that also in Italy, there is a lack of manpower, lack of people that want to work into the fields. So, my answer is easy. So, I believe that this kind of farming is absolutely one of the coolest things that you can do in your job. Because as a student, studying every day and looking for more information, more activities, more research, this is exactly what precision farming is about. So, I believe that it could be really weird that if a student, after all the study of agronomy, after it, when he has to start to work, will leave it. Because this is really motivating. First thing, so the technology for me is really motivating. But on the other hand, it means that you have to apply. As I said before, without people, there is no technology. So, people are mandatory to run these kind of things. And this thing is here, but it's also in Italy, like in Spain, like in whatever. Guys, the motivation is something that first has to come from responsibility. We are talking about food. If people are not thinking about food as a something that is mandatory for life. There is something that is missing in let's say, in the society. The society should really spend more time talking about agriculture, what is really agriculture, and make the people, the young people, understand why it's so important. So, technology is cool, technology is fine, but you need people. And second of all, remember that without agriculture, there is no food. So, I believe it's quite motivating. I'm glad there are no more questions.
Just a simple one. Thank you for excellent presentations. In Kiamuri Farm, where we are trying to start with introduce the precision agriculture as a practice, especially in Europe, what are the research problems? What are the issues that some of the students who are here and masters also have to do projects, thesis and all the rest. What would you see in terms of how we can involve and can work together between the university in terms of these research issues and Kiamuri project?
So very good question. Very good question. So the question is, if I understood well, what the university can do with Kiamuri Farm and what the students. So again, students are the base of the success. I truly believe that a student that has the opportunity to work at the Kiamuri Farm is a very, very lucky student. So they have to understand that the university have to understand that this kind of situation, so as you see, we are not talking about a big farm, we are talking about a canyon farm really, really connected to the farming ecosystem, the small farm ecosystem of this land. To have students from university goes there, take their expertise to the farming school. On the other hand, have a farm that could give back some technology and expertise to them is a win-win situation. So I believe that if you send a few students to make an internship, that is fine. But I believe that you should organize, the university should organize some, let's say, journey there, 1 or 2 days to see what are the operations, what is the equipment, what is the equipment. Talk with the people that are working there, like Luisa, and understand how it works, because there is a lot of other things that they can learn. And then, last but not least, you see that this picture is a self-explaining. So don't think that technology is hard to understand. Louise mapped the field, so make this map after 15 minutes. It's a quick job done, but it will give her and to the farmer a lot of information to work with in the rest of the year. So I believe that Meru and the Kiamuri must work together in a win-win situation. I don't want, and I believe also Gianfranco and nobody from the friends that I have here, wants that the Kiamuri Farm is a place where to send students just for fun for a day. Must be a cooperation to make the university and the Kiamuri Farm successful. Thank you, students.
Thank you. Thank you so much. I was just wondering, other than shipping our students there, if the same can be replicated here. We have a very rich land here in the university, and I was wondering, why can't we have this farm in our own university here, so that instead of us coming, all of us cannot come there. I also have a small farm at home. I want to practice precision agriculture. If I see it here, it's easy to learn from here. How possible is that? I was just thinking aloud.
She has the time to answer.
Yes?
Can you second the question?
It was just one.
What was the question?
It was more of a comment, but you can respond. I was just wondering because it's like we will be having our students coming there to learn if there is that possibility.
But then what will be the limitations of starting the same kind of farm, the same kind of technology, that through collaborations it can be done here, and it will help many others.
Yeah, of course it can be done everywhere. Starting from university looks obvious. So I believe it's a good idea to if also the student can work directly from the university here and start to experience something into the boundary of the university. Don't go there. So my suggestion is to guys, let's speed up, let's do something within here, let's move up. So I always make maybe a not very nice joke about African time, but let's speed up the guys, let's move it and try to do something within these boundaries, not always ask to someone else to support. I believe this is a great university. Professor Odiambo told me that 7 years ago there was 44,000 students, now there are 16,000, correct? So it means that this organization is super powerful. I believe it's enough. You have the strength to make it happen.
Is there any technology you think that...
Yes, of course. We can definitely talk about and discuss if the Mary University can use it, but of course it is possible. Just to go and do it.
Okay. Thank you, our DVC. Please welcome.
Thank you. Let's appreciate Matteo. We want to appreciate him with a gift. I'll be ushering the Vice Chancellor to come. Susan, you can prepare it. But my comment was just one, and the school is here, the School of Agriculture. Just like Dr. Simiyu is saying, other than students going to study, I know in Italy, and our family, our friends from Italy are here, there is aging population, especially in Europe. How about our young students as going to work there for short durations, like a year or 2 years, and then they come back? Just something that you can think about and the school can also think about. So I want to welcome our Vice Chancellor here to award Matteo for the wonderful presentation he has made here today. And I think I can see the gifts is engraved the name of Matteo Antonella.
Thank you very much. Thank you very much. Thank you very much.
We really want to thank you for being very faithful friends with the university, and you are still welcome with our colleagues next year. But this time you come also with your wife and your daughter.
I will. But don't forget about my friends that I'm here just because of them. So all organization and everything.
Thank you.
And can I just give you one thing, because I believe it's the professor. Hold on a second here. I also have a gift for you. Surprise. So just so it's a smaller one. Just to understand, so last year when I was here, I started talking with the Professor Odiambo, and Mr. Odiambo and Professor Odiambo, and we have the lucky that we in Italy are working with farms, but also with other universities. So last year, Professor Odiambo asked me if I knew some other university that they can work with. So maybe as a standard presentation with the University of Padua, I told them about this opportunity, this connection that I have with the Med University. And you started, and they started. So the university started a big project, international project called Horizon. Last week I was visiting the University of Padua, and they told me, I didn't know, actually the project was going on, and they are expecting some good results. So these are very good news, because the University of Kenya and the University of Padua are collaborating. And so to rate this, the University of Padua gave me this. Now I can give you. You can open. So it's just a symbol of their friendship that they have with you. So this is a medallion that talks about the University of Padua. This is the drawing of the
with a beautiful lady here.
Yeah, I believe it's the same. There are a lot of beautiful ladies.
So we thank you so much, our Vice Chancellor and your team. We appreciate the presentation. Shamlik Mwangi is supposed to be the next presenter. And please, I had requested for questions, for those who had questions to submit to for earlier presenters. I have not gotten any myself. And I'm not sure if my colleague Loifo, Dr. Loifo, you have any questions that have been submitted. Because we have some early presenters and we have a new team. I think we were all here in the morning. Anyone with a burning question to those who are with us? And those who are with us, I hope you are still with us. Si una present watu wanasikiza yako na wanapotea. This is not good manners. Where are Jen? Oh, I thought you had vanished. Thank you. Thank you for showing up for the afternoon session. So, members, it is interesting that no one has a question. Okay. Which presenter do you want to ask? Lucy. And
they can come here. The presenters can come here so that they can answer the questions.
You can come.
So very quickly, as she comes, I just had one question for Anne. The question is this: I saw your presentation was quite good, but of course, because of time, you went very fast. But my question is, you are looking at a comparison of the commercial fish meal versus the one that you want to formulate with the BSF. And I just found that you are a little bit biased on just looking at the protein content. And there is something striking that probably you should actually also look at the ash content, which actually helps. It is like the higher it is, it causes some indigestible. It is like an indigestible filler. Okay, so if it is in very high concentrations, again, you might think that you have good protein. But due to the high ash content, especially for the fish meal, you might find that it is not being taken up the way it's supposed to be taken up by the fish. So instead of just concentrating on that, you don't have your presentation there. I wanted you to go to the table and you just give me
your thoughts about that, because from the fish meal the ash content was quite high, but in your formulation the ash content decreased. And the last one is, I would have loved to see a pictorial, to see the different sizes of this fish, which are fed by the fish meal, and now are there any differences in terms of their sizes? And so can you comment on that? How does the ash, because you are just looking at the protein, and you do not look at it holistically in terms of all the components and how they can influence each other. So how does the ash content influence the uptake or digestibility of the protein?
Okay, thank you very much. Yes, Dr. Gray, the study found that the fish meal is superior. It has more ash content, and the ash is related to the minerals that are necessary. So that is why we are not going for full substitution. We look at the diet 3 that had the 100% substitution for the fish meal. It was relatively low in the ash content, and hence that is why we are not going for the 100% substitution. So we still want to remain part, so it is partial substitution actually, part of the fish meal and part of the black soldier flies. But we still have that balance of the ash aspect. And again, also when it comes to the non-digestible aspects, from the food fiber and the food fiber related to the chitin or the chitin, again, we are not going for the 100% substitution. We still want to retain part of the chitin and have less of it. So the 50% substitution gives a better balance. Chitin, we also don't want to go to zero part of it, because it is also essential in the antimicrobial properties. It still have more advantages. So that comparison. So we are not going for the 100% substitution. We don't want to do away the fish meal completely. We want to have a diet that is having part BSF, part fish meal. I don't know whether I've answered your question. So maybe I didn't have enough time to also have the discussion against fiber aspects, the ash aspects. Thank you.
Okay, let me have it. Yes, I have a small question about the BSF. You said it's cheaper. Yes. Out of experience, for you to rear BSF to have the protein you wish to have, you require another source of protein.
Yes.
And that's why it has been a challenge in adoption of BSF as a substitute of feed to fish meal at the industrial level. Even if you are using human waste, I was trying to think, if I want 10 tons of human waste, after that I will never get another waste of that magnitude. If I get 10 tons in Meru County, I will have emptied all the toilets in Meru. After that, I can't get another 10 tons to rear BSF. So where will I get the waste that can provide that kind of protein, other than the human waste? Thank you.
Okay, you may not require the 10 tons actually, because the protein sources can also be from other different sources. The food remains, we still have part of the proteins, the food remains, we still have certain percentages of proteins, and actually we will not require a lot of the 10 tons, as you say. A few urine diverting toilets will give you necessary proteins to mix with your other organic waste, and you'll have your BSF. Those studies have been done earlier. That is why this study did not go into the details of producing the BSF. Actually, they have been done in the Sanitation Institute. Coming back to your question, we had talked about the behavior or rearing of the fish, actually. So this study also did the rearing of the fish. in a complete randomized design. The setup was still in SRI, the Research Institute, and we looked at the growth, performance, and the economic aspects of the whole process. And even if now there were slight differences, marginal differences registered in the commercial diet, very marginal to the one that is in diet 2. If you look at the economic performance, then the trade-off because of the return or investment aspect still makes that diet of partial substitution of 50 to 50 a superior diet. Thank you.
I had a question for you. I might leave it those questions because of time.
Yeah, thank you so much, isn't it? Yes, in the morning you presented very well, but I had a concern because we know human waste is associated with heavy metals. I think I asked this question again in the last conference and I wasn't satisfied with the answer. How safe are we to consume a fish which has been reared using proteins from BSF, which have come from human waste, or the waste we talk about? In fact, most kitchen and other waste have heavy metals. Because we know organisms are very good by accumulators of heavy metals, especially fish. Fish, you give it a gram today, a microgram, within a very short time, it has been accumulated. That's why even pregnant mothers are not advised to take fish because of that. Now you feed fish with an organism which has been reared using the waste. Fish is very smart in accumulation. What happens? How safe are we? Have you ever done analysis to ascertain that the BSF that we use to feed chicken, fish, there is no presence of heavy metals, or just do the approximate analysis that you showed us there? Thank you.
Thank you. The study started by also looking at the microbial contamination and the metal contamination of the BSF that was produced. I agree with you that heavy metal contamination will still remain, but the microbial, the BSF is able to fully eliminate the microbial aspect. So heavy metals, again, we will say that let us have careful also selection of the waste that we want to use and the intended purpose. So if I'm rearing BSF to be used for feed, then the choice of where I am collecting my organic waste. If I'm rearing BSF for remediation of the metals, then I'll use the BSF to remediate the metal aspect and maybe not use it for food. But for this study, we started by looking at the waste that the BSF that was reared. It was free of the microorganisms, it was free of the metals, and again we also did a confirmatory test on the fish that was produced. They were still not there. So a choice of the substrate also is essential. Thank you.
I think those are enough questions for her. There were other presenters. We cannot be converting only one person. Others also need to feel the heat of being in a conference. And who?
Lucy Gasheri, I had that opportunity.
Lucy Gasheri, are you here? And Toroka. Madam Jane is here. I think she was presenting in my area. Can I think of something for her? Oh, you, Madam, you are talking about momentum equation. Am I right? Oh, can we skip class kindly? Yes, she was talking for physics. She was actually, I believe it's applied mathematics, right?
Yeah.
Yes, I did a double maths applied option. So we are in the same field. Now you are talking about momentum equation, which of course is also featuring in physics. I would like to, have you ever heard of Noether theorem?
No, I didn't deal with it.
Okay. Now, Noeda theorem is about conserved quantities. Any conserved, which are the conserved quantities you came across in your research?
Mass conservation. We are talking about the equation of conservation of mass, the equation of continuity. It is the first equation I talked about. The conserved species is mass.
Okay. Yeah. The Navier-Stokes equation?
The mass momentum equations are known as the Navier-Stokes equations.
Yes.
Yes.
You talked about it?
Yes.
And you dealt with it because I saw it on the board?
Yes.
Now, among the conserved quantities you've mentioned about mass, what are the physical significance of those conserved quantities and any assumptions you may have had while actually getting those kind of results for the conserved quantities?
In our equations, we were interested in the flow variables, and we were combining the mass conservation equation and the momentum equation was giving us the flow profiles for the velocity field. We were interested in the velocity field, how the velocity profiles behave in the enclosure that you are dealing with. In the energy equation, we were interested in the temperature profiles. So our study was basically on the profiles.
Thank you. Thank you. You can take your seats.
Thank you.
Please, thank you so much. I want to have, we rearrange this one for those who came late. I'm being reminded that some of you may not have sent on the email your work. Those who have come late and did not send in the morning, please ensure that you have your work sent on the email. Otherwise, I would want to invite Jamnik Mwanki, you are here? And you are ready? Please, you are just a moment. I don't know where my other Actually, it's unfortunate making consultations because of the time. You know, according to my programs, I was giving you 10 minutes, working including the time we had for our visitors, that we were going to almost around 6. We will be done. But now, Mukeli Joe Mwenyeji Apone, what do we do? If I give him 10 minutes and the remaining 10 minutes, let's be honest, you don't need to be a double mathematician to do this. Looking at the watch, that will not be visible. And we do not have any other opportunity for you to present, and we want all of you to present. So I'm feeling like cutting short your time to between 5 and 7 minutes. You might have to shorten further. It is not our wish, but you know we could not block our visitors. They had to be with us. They have traveled all the way. So please, I would appreciate if you finish your work in 5 minutes. But if so long, let's not exceed 7 minutes. Thank you.
Okay, thank you very much, professors, doctors, and protocols of staff. Welcome to this presentation on the mechanics and the statistics in the field learning. It's a review study, and my name is Mwangi Jandrek. under my professor, supervisor, who is Professor Gitonga Ganeta, and I'm very grateful for his work. Effluent bioslurry is generated as a byproduct of anaerobic digestion, which happens during the production of biogas. And in this, the outlet of biogas contains the bioslurry itself, which can be carried microbial as well as residual and methane. The expected is that the bioslurry anaerobic digestion will be very efficient in terms of conserving the energy as well as not letting any microbial or any methane leaving the digester. But due to other factors, some of them escape and they go to the displacement tank as well as the slurry tank. The process of methane production follows four main processes, which include the hydrolysis, which is about the turning of complex material into simpler soluble materials. Then it goes to pathogenesis, now the conversion of the volatile fatty acids, long-chain volatile fatty acids, into short and acetate. That is now the second level, of the acidogenesis. And finally, the metagenesis process. And all processes are at the control of microbial populations in different abundance as well as different composition. So, for this review study, the problem statement was on that digestate is what gives the digester. It can contain microbial biological processes which enhance methane continued production during the storage and transportation as well as application time. With this, it contributes to the greenhouse gas footprints. And for this justification for the study is that we need to understand the potential of methane to quantify the environmental footprints in the biogas systems. The objective of the review was to review the composition and the abundance of microbial communities present in the digestate, and also to review the methane content in the bioslurry. We used a systematic approach and we reviewed 44 peer-reviewed articles and also a case study that were published between 2012 and 2026. And the results were at the residual methane and treated microbes is that there was no explicitly literature that was quantifying the methane content in the digestate. But for microorganisms, we found different categories of microbial population that were found, which included both the beneficial arachne, the beneficial bacteria, the fungi, as well as residual pathogens. The different bioslurry types also had difference in terms of the residual methane potential as well as relative microbial loads. And this was characterized by different load in terms of the organic matter that was used. For instance, from the pig's manure, bioslurry had the highest compared to the crop residue, which had the lowest or the low level of methane potential due to its high level of lignocellulose as well as hemicellulose and lignin, which limit the biodegradation. The composition of putative microorganisms in the bioslurry is that firmicutes had the highest in terms of the taxa, and this is because of formation of spores, which prevent them to undergo further digestion in the anaerobic condition. And the lowest was with the Ascomycota, the fungi, at 5%. In terms of the genera, Clostridium had the highest, though it didn't give the values in terms of high, moderate, and low, and very low. So we have the Clostridium, the bacteriocytes, Luminicoccus. Those are all bacterial family. Then we have the arachne family. We have the melanocyte and Melanopsamica. These ones are able to continue digestion of substrate in the storage level. So, what are the impacts of this microbial population in the biosphere. One is that they enhance continuous production of methane, which is a greenhouse gas emission, important in carbon footprint. It also enhances the residual methane to reduce the environmental benefit associated with the aerobic digestion system, meaning if you have higher level of microbial due to high level of organic matter or substrate in the digestate, to result also in high methane production post-digestion in the digester, which now results in more greenhouse gas and fluxes to the environment. Another impact of the residual methane is that it reduces the energy recovery efficiency, which now is supposed to be part of the work of the biogas in terms of climate change mitigation. The impact of the positive microbes is that their presence indicates incomplete digestion in the digester. Also, when they escape the digester, it means that they contribute to microbial communities, ecological balances, contributing into environmental contamination. And finally, the high microbial loads can limit the safe and occasional use of digestate as a fertilizer. And we know that biosphere is one of the nutrient-rich compound that is used as a biofertilizer. For the conclusion is that digestate remains a biological active product of anaerobic digestion, which retains high level of microbial communities, which enable continuous methane production potential. Without the case, then they need to improve the monitoring and management of biosphere is very critical. And with this, to enhance resource recovery, while minimizing the greenhouse gas emissions and the microbial risks. And for that, we have a commission on having further research on methane, on residual methane.
Thank you.
Thank you.
Thank you. Thank you so much. Let's appreciate him. So our next speaker, I said Chamlik, then the next one should be Oliver. Then you will be followed by Eldon Mwereki. Eldon, are you here? Yes, in that order. So Eldon Yakitoka, then we shall be moving to William Mathenge. Then maybe after that we can have some questions. How much is work and Musho when all of them are? Yes. Yeah, because you have to wait for the rest. I would have been on the end of chair present.
Oliver is not here. Oliver is not here.
I've not seen him. Oliver is not here?
No, I haven't seen him.
He's not.
Move to the next person.
Interesting, but I've already called the order.
The supervisor is there.
Yes.
Dr. Cynthia. Linda Gago.
Where?
But from Germany we want to go to Oliver and then we go to Elton.
The supervisor is, the supervisor is there. You can ask.
Who is the supervisor?
Cynthia Mugo.
We are asking where is your student? Okay, where is he? Do you have any information?
Let's go on. He's not here.
He left. You are expecting him back?
I don't know whether he's back.
Okay. Okay, so let's go with those who are present.
Yes.
Elton, you ukisikia mwenzako naenda na hajaamka, you are the one next week. We don't have that luxury of time.
So good evening everyone. Welcome to my presentation. It's based on the evaluation of cement handling practices in Kenya, and I'm the author, and my co-authors are Joseph Mwangi Kaigai, Victor Kipto Mutai, and Dr. Joseph Mwiti Maramu. So for my introduction, I'll first of all introduce cement. What is cement? Cement is one of the most widely used construction material, and it's the main binding material in the making of concrete, mortar, and precast. And on the global scale, you have noticed that the cement demand has grown highly because of one, rapid urbanization, industrial development, and infrastructure. Specifically in Kenya, we've noticed this growth attributed to government initiatives such as affordable housing program, rapid urbanization in both peri-urban, urban, and rural areas. And as from the data shown here from the Kenya National Bureau of Statistics Economic Survey report, as of 2025, the production of cement in Kenya was 10.4 tons, and the consumption was 10.3 tons, showing that as much as you're producing a high scale of cement, it's also being consumed widely internally. So I'll go another link to explain the problem which you're facing in Kenya currently. You find that cement is hygroscopic in nature and it's prone to environmental factors. These environmental factors are many. One is moisture, one is humidity, and the factors which mainly contribute to quality deterioration of cement are high humidity, moisture ingress, temperature fluctuations, damaged packaging, and prolonged storage. These factors tend to happen in the construction supply chain. And by the meaning of construction supply chain, I'm referring from the manufacturers all the way to the end users. So you find the construction supply chain, we have numerous individuals who are handling cement from the manufacturer all the way to the end users, who is usually either the contractor, the artisan, or these local fundis. So you find from the manufacturer, we have the distributors. The distributors are the ones who are now packaging this cement into lorries. Then they're delegating this off to the transporters. From the transporters, it goes all the way to the hardware shops or the construction sites or the warehouses. Then from the houses, we have the contractors or the artisans. And finally, the end users, for example, people are now using this cement to make their products, for example, inside their own home sets. So, to my problem statements, as I said, we are facing one critical challenge. As soon as the cement leaves the manufacturer, the quality has already been fine because you already have a standard called ES 18 per ton 2017. So you find once this cement leaves the manufacturer, it has met all the quality requirements. But the quality at the end point of use cannot be guaranteed because, as I've said, the construction supply chain is long and we have numerous individuals. So you find these people tend to have an effect on the final quality of this cement. So you find when this cement has been tampered with, this improper handling results into 4 factors, which is prehydration. This is when cement reacts with atmospheric moisture, carbonation reaction with carbon dioxide, then material wastage, because you might find this bag has been already damaged, then quality degradation. For example, if not the quality of the cement has reduced, you find that your structure may build a structure but is now subject to crumbling or internal stress because of the damage to its quality. So for my objectives, they were as follows. One was to identify the common challenges associated with cement handling. The second one was to determine the level of compliance of stakeholders. The third one was to propose practical measures for improving cement quality preservation and minimizing losses. So for my justification, why does this study matter? As I've shared, cement demand continues to grow. So you find, even for the next generation, if we don't handle this problem, it will go further to impact even the forthcoming generation. And why am I focusing on this? If we improve the quality of cement by preserving its integrity, one, we will cut down material losses, we'll be able to spare construction costs, and we'll also be able to reduce quality failures. And from this study, I'm expecting to assist manufacturers in identifying post-distribution quality risks. The other one is support policymakers, by this I mean KEBS and the National Construction Authority, in developing guidelines and awareness programs, improve construction quality and durability, contribute to the local knowledge on cement supply chain management, and also contribute to SDG 12. This is responsible consumption and production by promoting resource efficiency and minimizing cement wastage. For my methodology, I used a mixed methods approach, whereby I delegated questionnaires to a number of stakeholders in the field, and I also proceeded to visit some of the retail points and the hardwares, and also took some photographs of some hardware stores which I visited. And from the data I collected, I subjected it to data analysis. And for the data, I had a number of 40 respondents, and that 3 of them were outliers. They didn't give compatible data. So from the data which I got, from that 7, majority of them were the individual consumers, and the others were distributed as shown there. And you find for the responding categories, most of them had the experience of 1 to 5 years, and several of them had beyond 5 years, even some going to 15 years in terms of the manufacturers. And now when I subjected them to the question of how do we receive their bags from the store, most of them rated them to be good, some excellent. But they also reported cases whereby they have experienced damaged bags frequently or occasionally. For the storage duration, I also subjected them to the question, and most of them reported that the cement is usually stored for a maximum of 1 month. Some of them said less than 1 month. Others said 1 to 3 months, and even some of them reported duration beyond 6 months. This, from the standard, it says you're not supposed to keep cement for more than 3 months. So it's showing there's a gap in compliance, because you find if somebody can keep a cement bag for more than 3 months and still sell it to someone, that means there's a problem somewhere, because most of the people in the country are not educated on how they should handle cement, and some of them are not even aware that cement usually has an expiry date. So as I've said, some of them reported 6 months. So you find the longer that cement stays in the store, it's now prolonged to be attacked by the environmental factors. So for example, if predation were to occur, this results to now formation of lumps in your cement. And as you can see, from most of them, they say they reported that they have experienced at least frequently, sometimes or rarely. But none said that they have never experienced lumps in the cement. So you can see this is a critical indicator that for most of the cement in the country, people have been exposed to now cement which has undergone quality deterioration. That's meaning there's a critical gap in understanding why is this happening in the cement supply chain. And for the other part, when I subjected those to another question, they said that they attribute poor handling during loading and unloading. You find that when people are unloading this cement, for example, the people are unloading, they don't know that they're not supposed to keep cement on the floor. It's supposed to be raised on pallets. So you find it undergoes, it's already starting to be subjected to environmental factors. And for the factors which people attributed mainly to the damage of cement, most of them attributed moisture exposure and poor storage as the highest factors, followed by these other ones. Then for my conclusion, I said that one thing, the conclusion that as...
Thank you, sir. I appreciate. Thank you so much for that good presentation. Please let's keep a clap. The next person. You should be, when you see me starting, you also be coming.
Thank you, sir.
That's William Madenge, right? After William, we will be having, I want to know if Nina, Nora, you here? Good. And then you will be followed by Bill Press. Yes.
Good afternoon. My name is William Madenge, a geologist and a second year master's student dealing with the topic of upcycling potential of quarry waste in the construction sector in Kenya, and these are my authors. So on to it, the background information. So the construction sector in Kenya heavily relies on the quarry-derived aggregates for mainly concrete, masonry, roads, and infrastructure development. So there are type of aggregates that do this work. So they are the overburden, reject rocks, the quarry dust, and the streaming fines. So once the quarrying activities are being done and the aggregates are being extracted, they leave the land degraded. There are effects such as land degradation, air and dust pollution, and unmanaged waste and impact visualization. So what is the aim of this study? This study aims to investigate how this waste can be upcycled so that they can benefit the construction sector in general, and it can be done by identifying the waste resources, then developing value-added materials, then they can be used in sustainable construction. So in Kenya, there are 5 main regions that contribute to the supply corridor of aggregates in Kenya. There is Kiambu, the Darugo that constitutes the biggest construction materials source. There is Nakuru, there is Meru, Murang'a, and Machakos. So these quarrying activities in Kenya are concentrated in these major production belts. So the problem statement here is that the quarry waste is widely generated, but it is purely documented and underutilized in Kenya. And the extensive quarrying across the major production belts in Kenya now leaves sites like that. It is a visual glow to see. So the current situation is that extensive quarrying along the major production belts in Kenya is being done, and the challenge is that the large quantities of quarry waste are accumulated and are not being used. So what are the consequences? The land degradation, dust pollution, visual intrusion, and loss of productive land. So there is also a knowledge gap as there is limited information on the spatial distribution and mineralogical characteristics of the resource. So this presents us with an opportunity where we could turn the quarry waste into value-added materials that could be used in cementitious materials, pavers and blocks, road base and surface, and concrete applications. The objective of this study is to evaluate the recycling potential of quarry waste in the construction sector in Kenya, and the specific objective of this study was to map major quarrying hotspots in Kenya and identify regions associated with significant quarry waste generation, to correlate the quarrying activities with the geological formations underlying major quarrying regions, to characterize the quarry waste from selected quarrying regions representing the dominant geological settings of major quarry waste area in Kenya using XRD, and finally to assess the suitability of quarry waste for value-added applications in the construction sector. So this study is in line with SDG 9 and 12, and also with Komayangu and Kenya Vision 2030. The methodology used for this study was number one, the reconnaissance done by the GIS mapping, then the geological map analysis, quarry hotspot identification, field sampling and data collection, lab analysis using XRD, and the mineralogical characterization. So the GIS was used to map the quarrying distribution, the geological maps were used to relate the quarrying to rock formations. Representative samples collected, we chose two locations due to time, but they represent the general geology of the quarrying areas, that was Murang'a and Kiambu, and finally XRD was used to determine the mineralogical composition. So this is a geological map of Kenya. The grey area is the Rift Valley that has the volcanic rocks, and the yellow area at the middle is the basement system rocks that also is used for the quarrying activities. So these quarrying hotspots are controlled by the occurrence of Precambrian rocks. Somebody might ask, why don't we have quarrying activities in the northeastern or the upper or the coastal? It is not random, it is dictated by the geology of Kenya. After we took our samples from the field, we characterized them using XRD. So the image there is a diffractogram. It is used to tell us what exactly the mineralogical composition is in the samples. So we have muscovite, nontonite, plagioglast, and snectite. All these are minerals that have aluminosilicate minerals, which will be used in the discussions here. So the discussion from this study showed that the quarry waste from both Murang'a and Darugo are dominated by silica-rich minerals and aluminosilicates. Yeah, so the high silica content enhances mechanical strength, hardness, and durability, whereas the presence of feldspar suggests the possible pozzolanic behavior, especially when finely ground. So the potential uses of this quarry waste can be in structural and non-structural applications, performance comparable to the conventional materials, promotes sustainability and reduces natural resource extraction, and finally it is cost-effective, environmentally friendly alternative. To the conclusion, so this study concluded that there is a clear relationship between the geology and the quarrying activities. The XRD analysis showed that the quarry waste from Murang'a and Darugo are dominated by the aluminosilicates. Then the mineralogical characteristics suggest that the quarry waste possess good potential for reuse in the construction sector. Overall, quarry waste studied in the region represents a viable secondary resource supporting circular economy and sustainable construction practices in Kenya. Finally, the quarrying activities in Kenya are strongly concentrated in central Kenya and the Nairobi supply corridors, reflecting the distribution of suitable aggregate-bearing geological formations. Recommendations. So there should be further testing, mechanical durability and pozzolanic performance to validate the suitability of different construction materials. We should develop a national quarry waste inventory and geospatial database to improve tracking, quantification and management of quarry byproducts. We should promote industrial scale utilization of the quarry waste in cement, concrete and masonry and road construction materials. We should also have policies in frameworks that encourage quarry operators and construction industries to adopt waste valorization practices. Finally, we should encourage collaboration between researchers, industry, and government agencies to support innovation in sustainable construction materials. That was my presentation. Thank you.
Thank you so much. My friend, you are younger than me.
They have learned how to innovate.
Good afternoon. So the title of my research study was Special Distribution of Biomass and Agricultural Waste-Derived Supplementary Sanitation Materials in Kenya, and these are my authors, Mr. Joseph Kaigai, Mr. Victor Mutai, and Dr. Joseph. So the background of my research topic is that Africa's urban population has increased tenfold in the past 50 years. That is from 32 million in 1960 to 448 million in 2020, with projections that the population will have doubled by 2050. From the graph there, you can see that the bar shows how there has been rapid population growth over the years, and the red curve shows the urban population, how it has been increasing over the years. So with the rapid population growth, this means there's going to be an increase in infrastructure, means there's going to be strain in infrastructure, and with strain in infrastructure, there's going to be more demand for housing. And for housing, the main raw material we use for housing is cement. However, cement has its limitations. That is, cement is expensive, and also it has CO2 emissions, that is from calcination, and from also the fuel conversion. So with this, how can we help now solve the issue of cement? This is by use of supplementary cementitious materials. These are materials which are used to partially replace cement, and these SCMs are rice husk, sugarcane burgers, coffee husk, water hyacinths, sesame residues, and coconut shells. All of these are agro-waste, and agro-waste are poorly managed, and mostly how are they managed? Mostly it's by open burning, and during open burning, the agro-waste they release particulate matter, they reduce the air quality, and they release methane, CO2, and they also lead to environmental degradation. And we can use these SCMs to promote the circular economy by using as construction materials. And why these SCMs? This is because of their pozzolanic reaction and the property enhancement. Also from the literature, it shows that these SCMs, they can reduce the clinker factor in cement by up to 30%. So the problem statement of this research was that for cement, the conventional cement, it's very expensive, and if we use SCMs, we're going to reduce the production cost of cement, and also for cement, it has high CO2 emissions, and also for agricultural waste, most of them are usually burned and discarded, and there's limited information on SCM resource distribution in Kenya. The justification of this study is that it justifies SDG goal number 9, that is promoting innovative construction materials. SDG goal number 11, by use of SCMs, we're going to support affordable and sustainable housing. It also justifies SDG goal number 12 by encouraging waste recycling. And also by use of SCMs, it justifies the Boma Yangu, which is the affordable housing program. By use of SCMs, we are going to reduce CO2 emissions. That is, we are going to take urgent action, which is the climate exchange. And also by use of SCMs, we are going to create more job opportunities. For objectives, the general objective was to evaluate the spatial distribution of biomass and agricultural waste, to identify biomass and agricultural wastes suitable for SCM production, to quantify the annual generation potential of these waste materials, to map the spatial distribution of SCM's resources using QGIS, also to assess the chemical and mineralogical suitability of selected waste as SCMs. So in this slide here, we get to see that rice husk, they are mainly in Mwea, Ahera, and Bunyala. Sugarcane is in the western Kenya and the Nyanza region. Coffee husk is in the central Kenya and Mount Kenya region. Coconut shell is in coastal Kenya. Sessile residues in Taita Taveta and Kilifi. And in this map here, we get to see the special distribution of this agro waste all over the country. And around Lake Victoria, we can see there is water hyacinth. And in the regions of Kakamega, Bungoma, there is sugarcane. And in the regions of Kirinyaga, we have rice. And the coastal areas, we have coconut shells. And Taita Taveta, we have sisals. So from the research that we did, we got to rice husk produces around 80,000 tons annually. Sugarcane burgers, it's around 2.5 tons annually. Coffee husk, it's 200,000 tons. Coconut shells, it's 50,000. Sessile residues, 300,000. And water hyacinth, 1 million tons annually. And also from this, the key, we found that rice husk, it has the highest silica potential, that is about 80 to 95%, followed by sugarcane burgers, which has around 60 to 80, and then it was followed by water hyacinth and sisal residues. And also from this, we got to discover that rice husk has the highest potential of being used as an SCM. This is because of the high silica content. So the conclusion from these studies that Kenya possesses that significant biomass resources for SCM production. Sugarcane burgers represent the largest biomass resource. We saw that it produces around 2 million metric tons. And rice husk, it exhibits the highest silica potential. And water hyacinth, it's around 1 million tons annually, but also it's not utilized well. And the high SCM suitability areas were identified, and also transport distance significantly influences SCM feasibility. So recommendation of these studies we should promote utilization of agricultural waste for SCM production. We should establish regional biomass processing center, prioritize rice ash in SCM development, formulate supportive policy frameworks, conduct life cycle and economic assessment, and also develop a national biomass resource database.
Thank you so much, madam. It's time. Great appreciation. You are finished anyway. And it's very good. Who is following next? We have Nancy.
Yes, so good evening everyone. My name is Bilfred Mwenda and I'll be presenting on the assessment of fine aggregate sources in the construction industry in Kenya, and these are my co-authors. So as we all know, is that fine aggregates are a major resource in the construction industry in Kenya, that is masonry, plastering, flooring and screeding and all that. However, we see that there is a rapid urbanization and infrastructure development in the country, which has led to growing demand of these fine aggregates. However, there is an increased, now the fact that there is a growing demand of these fine aggregates, we get that now there is increased demand, which has led to now questions arising on resource depletion, utilization practices, stakeholder perception on transport, cost and distance. So to the problem statement, we ask ourselves what is known We are talking of what we know about the fine aggregates are the strengths, the performance of these fine aggregates. But there is a limited socioeconomic and stakeholder-based information that exists between these, about these fine aggregates. To my justification, we see that this study seeks to bridge a gap between now the technical knowledge of fine aggregates and the stakeholder decision that influence their sourcing and their utilization within this, the Kenyan industry. So to my objectives, the general objective is to assess the source selection factors, cost and utilization practices. The specific objectives is to identify the major fine aggregate sources, to determine and evaluate the factors influencing aggregate selection, transportation cost implications and utilization practices, and then to also evaluate the perception towards alternative aggregates in the construction industry. So, onto the methodology, I adopted a descriptive survey design which targeted the construction industry stakeholders. These are engineers, the aggregate suppliers, the regulators, and other professionals, including researchers. Data collection was primarily collected using questionnaires, that is online questionnaires. Then data analysis, descriptive statistics were used, that is on the using frequencies, percentages, mean scores, and now to the results. First we see that a good number of approximately 32 respondents which included the aggregate suppliers, the contractors, the regulators, and all that. So from here we can see that river sand still continues to dominate as the major fine aggregate source. However, some other people have used mixed sources and have also ventured into manufactured sand in the construction industry. We also see that the selection factors, the major selection factors affecting the construction, the picking of the fine aggregates, the quality and the cost. And so from the results we can say that aggregate selection in Kenya is primarily driven by the performance and the economic factors, which supersede other factors such as the environmental concerns, such as the land degradation and all that. Then to the transportation cost, we can see that most of the stakeholders confirm that transportation cost and transportation distance affected on the overall cost of the fine aggregates. So we can therefore show that it can confirm that the proximity to which we are sourcing our fine aggregates affects the cost. So the lower the shorter the distance, the shorter the proximity to the fine aggregate market, or rather the source, is now leads to now a lower cost. Then we see that that also the stakeholders have some have used manufactured sand, and some are venturing into choosing manufactured sand. Manufactured sand, for those who may ask, are these probably granitic or basaltic rocks which are crushed and graded in the industry and then sold and used as fine aggregates. So they have seen a gap where there is a fine aggregate depletion. So they have ventured into manufactured sand, and we can see that some are also willing to now take manufactured sand over river sand, which is being depleted. So then we say that in conclusion, we can conclude that river sand remains the dominant source, and we also say that material quality outweighs the environmental concerns. We see that also transport cost and distance significantly affect the the aggregate cost, and we also see that the industry is gradually transitioning to different sources of fine aggregates other than river sand, we are, the industry is also transitioning to things like the manufactured sand. And also now to the recommendation, we say that we should now promote quality assurance to ensure that there is a reliable and consistent aggregate performance. Then we should improve supply chains to enhance availability, reduce cost, and ensure timely delivery. Then we should also increase awareness of alternative uses of fine aggregates such as manufactured sand, tile waste, and all that. Then these actions can enhance sustainability and long-term resource security within Kenya's construction industry. That is the end of my presentation.
Thank you.
Thank you. So can have that's Nancy. Good.
Like they really have a little time because their presentations are so short.
So good evening. My name is Nancy, and I'll be presenting on durability concerns in urban construction. Welcome. So the construction industry contributes to 37% of the global and related CO2 emissions, and this puts the construction sector to some pressure to reduce on the environmental impact and adopt sustainable and affordable housing building materials which require low carbon and are locally available materials. Other construction materials, including the stabilized soil blocks, we have rammed earth, are potential sustainable alternatives as compared to the conventional masonry materials. However, their long-term performance, or rather the durability, is often compromised by the intense moisture-induced deterioration. Now, one of the biggest challenges facing the earth and construction materials is their vulnerability to the moisture exposure, which leads to reduced durability. And when these materials are exposed to this moisture, it leads to high water absorption, we have loss of strength, we have rainfall erosion, we have cracking, as well as repeated wet-dry cycles. There exist several mitigation strategies. However, there still exists limited information on these sustainable bio-based hydrophobic additives that are capable of improving moisture resistance as well as improving on the long-term durability of these urban construction materials. There is also lack of local relevant data and stakeholder insights on the effectiveness, potential and adoption barriers of these bio-based hydrophobic additives. This study is very important for several reasons. First is on the sustainability aspect, whereby it encourages on the utilization of the renewable bio-based resources and reduces the reliance on the synthetic admixtures. Second is on the environmental benefits, that is on the potential to lower the CO2 emissions of the construction and promote green building. That is on the performance improvement, as it is expected to reduce on the water absorption and moisture ingress, thereby enhancing the durability. Fourth is on the socioeconomic relevance, as it is expected to lower the cost and promote the utilization of the locally available materials that support the affordable housing. And lastly is on the scientific contribution, whereby it generates the new knowledge on the bioadmixture for urban construction materials. Now, generally, this study supports the development of the sustainable and low-cost construction materials that are very suitable for moisture-prone environments. The general objective of this study was to evaluate the durability challenges in urban construction and assess the current mitigation strategies which emphasize on the potential of the bio-based hydrophobic additives. And for the specific objectives, number one is to identify the major durability challenges affecting these urban construction materials. Number two is to assess the moisture-related deterioration mechanisms and their impact on the performance. Number three is to evaluate the effectiveness of existing mitigation strategies that are used in practice. Number four is to determine the stakeholder awareness and perception of these bio-based hydrophobic additives. Number five is to identify the barriers to adoption of the bio-based hydrophobic additives. And lastly is to recommend future directions for research and industry adoption. For the methodology, stakeholder surveys were adopted, whereby the key stakeholders which were put into consideration were the engineers, the contractors, researchers, manufacturers, as well as practitioners. And for data collection, structured questionnaires were prepared, which focused on the durability challenges, moisture-related problems, mitigation strategies, bio-based additives, as well as the adoption barriers. For the data analysis, the raw data was collected and descriptive statistics were used, whereby frequencies and graphs were adopted. Now moving to the results, one of the major durability challenges which was identified by the respondent was on the water absorption, which was found to be at 58.3%, followed by loss of strength. We have rainfall erosion, we have cracking, and the list was wet-dry cycle, but which is also very important. For the moisture impact, the frequency of the moisture-related problems was found to be very often or often by over 75% of the respondents. And when the respondents were asked about the most effective mitigation strategies, cement stabilization was ranked the highest, which was at 54.2%, followed by the waterproof coatings, hydrophobic additives, followed by the surface sealants, followed by the improved drying practices, and the list was on lime stabilization. So the cement stabilization remained the most preferred durability enhancement. On the awareness of the bio-based hydrophobic additives, moderate responded that they were aware of these hydrophobic additives. However, despite their moderate awareness, they still believe that these hydrophobic additives can improve on the durability. The major barriers to the adoption of these bio-based hydrophobic additives, we had limited research evidence, which was found to be 41.7%, followed by the lack of awareness. We have lack of standards, high cost, and list was limited availability. The most encouraging finding was on the support from the stakeholder for the research and adoption, which was found to be at 91.7%. In conclusion,
Julius Shoki, can we please appreciate her? Julius is here.
Yes, he's coming.
Oh, sorry. No, take your time, I understand.
So after 2 years, we shall have Frederick. Are you here? Frederick Bett. Yes, we will follow 2 years without being called. Yeah. And then participate. We shall have this guy, Douglas. Douglas, you're here? Yes.
Good evening. My name is Julius Musioki, a second year master's student, cement and concrete, and I will be presenting on evaluation of locally made standard sand, which is a shift from imported sand. So, in Kenya, we have at least 14 cement manufacturing companies. I know you can name at least 6, and each ton of this cement produced by each company has to undergo testing to conform with the construction standards. And one of the most critical testing parameters is the cement strength, which is the flexural and compressive, to confirm the strength which the load bearing capacity of this cement. So the problem, cement testing, according to KSES number 148 of 2017, the corresponding EN standard, it's the most important part. That sand is the most important part in this testing. And the smallest company will require at least 3 kg per day, and the largest company, which I know you can mention it, requires at least 28 kg per day. So the question is, where do we get this sand? The soap is, we import sand. Sand we import it from developed countries like Germany, France, and China. So, sorry for that. Yeah, no, the problem is, to import at least 1 ton of cement, you require 270,000 per metric ton, that's before taxes. And this will expose you to laboratory, will expose your laboratory to international supply chain handles. Every one of you is aware of the Strait of Hormuz. If that happens to sand, you will not test your sand and your cement, and it will result into poor quality materials. And the justification is in 2023, a study showed that Kenya imported 342,000 USDs of sand, and these imports create a gap in our net GDP. So the objective of this study was to conduct an evaluation of locally processed sand for use in cement strength testing. And the first objective was to collect, then you determine the chemical composition and loss of ignition, and the study also used this sand to conduct a test and compare it to the standard sand that is imported. The methodology was sand was collected from Mashuru in Kajiado, which is within the Mozambique belt, and the characteristics of this Mozambique belt is mainly the gneisses and the quartzites. So the sand given by such belt, it's highly angular and results into high interlocking. Then this sand was washed in the laboratory to get rid of silt and organic matter. Then we have your wash, we had the wash sand there. The sand was oven dried at 105, and the dry sand was then graded through the 2 mm sieve, 1 mm, 0.5, and 0.075 mm sieve. Then you obtain the residue on 1 mm sieve, the residue on the 0.5 mm sieve, and the residue on 0.07 mm sieve were blended in ratio 1:1:1. This is according to the current IS standard which governs this sand manufacturing. We have a picture of the standard sand that is manufactured by the Sinoma, and then that sand was exposed to loss of ignition through digesting it with HCl and igniting on a amorphous furnace. And yes.
Sorry to interfere. Is that result and discussion?
Yeah.
Okay, continue.
Then it was ignited in a amorphous furnace, and the loss of ignition was found to be 0.2%. Then, to continue with the results, the silica content of the sand was found to be 97.45%, iron content, and the alumina content. Then, the compressive strength was done for 2 days and 28 days. The reason is because the cement used for this testing was the 74B, and class 2.5R, which is required to be tested at 2 days and 28 days, not 7. So the strength at 28 days for the standard sand was found to be the 2.8, and at 28 days for the processed sand was found to be that 1.8. So according to KS number 18 of 2017, the conformity of standard sand, like of the conformity of cement, at 28 days, it should give you that 2.5 megapascals plus or minus 2. So you can see our process sand is within that range of plus or minus 2. The conclusion is that from the result, we can confidently conclude that the locally processed sand is viable for use in cement testing. Performance-based standards should be replaced by the prescriptive one to facilitate the adoption of locally produced sand. Like, tell me what to do and not how to do it. And then a clear mapping of sand in Kenya to create a...
Kennedy. Kennedy, are you?
Okay, good evening. Yeah, my name is Frederick Kibet and I'll be presenting to you assessment of dry mix mortar industry in Kenya. So, construction sector keeps growing in Kenya, especially because it's a developing country in Africa, as evident there. But the construction sector in Africa and especially in Kenya is also dominated by the skilled and semi-skilled labor, as you can see that demonstrated in that photo. One of the components that is used in construction is a cement mortar, and a cement mortar is a composite material of cement, sand and water. But this cement mortar is mainly produced at site, and this site-produced mortar has some drawbacks, one of them being high consumption of cement, limited uniformity in the produced mortar, and it's also labor intensive because you really have to hire people to do work for you. This also has low quality, and low quality results in drawbacks such as wall spalling and cracking of the walls. This leaves us with an alternative solution, which is the adoption of the dry mix mortar. A dry mix mortar is a composite material which has pozzolanic Portland cement, a graded sand, additives and admixtures. Additives such as the limestone, admixtures such as the cellulose, that is methyl cellulose and organic dispersible polymers. These are produced in the industry and packaged either in 25 kg, 20 kg or 50 kg bags. Some of them include the non-structural plastering mortar, tile adhesive, masonry mortar, flow screen grouts, and flow screen and powder skimming coats. Now, the major advantages for these dry mix is that it's a ready-to-use product. You really need to just add water and proceed. It's cheap, eco-friendly because the cement used can be controlled. Quality product, which is having more advantage compared to the site-produced cement mortar. But there is a limitation to this, by the fact that the dry mix mortar products, there is limited knowledge on the same about the dry mix mortar products. One person could use plastering mortar for masonry work, or masonry work for plastering, which brings in confusion. There is also limited knowledge about this dry mix mortar market potential. This forms the basis of my problem statement, to do a social survey on dry mix mortar products, market potential, and barriers to the growth of these dry mix mortar products. So this is justified by the Kenya Vision 2030 and also fills the gap on dry mix mortars. There is a focus to reduce the CO2 emission due to the efficient use of the cement. And again, lastly, market potential for these dry mix mortars. These are my objectives, from to determine the current level of adoption of the dry mix mortars to providing recommendations for dry mix mortar industry development. This started with this is my methodology, from literature review to doing the recommendation. This survey was done on manufacturers, distributors, standard bodies, and the consumers and end users. So from literature, globally there is already adoption of the dry mix mortars, but in Kenya there is only 55 countries, which 55 companies registered in the Kenya Association of Manufacturers, but these companies are only dealing with building and mining. So there is insufficient data on the number of dry mix mortar manufacturing companies that has been published in Kenya. On dry mix mortar construction materials, the distributors and manufacturers responded that these are the main products that they are dealing with in manufacturing, that is from tile adhesive to masonry mortars, tile adhesive taking the lead at 40%. Once they manufacture, the consumption rate, if a distributor stocks these dry mix mortars, it stands at 50% consumption, that is the purchase of these products. It is affected by a number of factors. One of them is the quantity in package. By the time a consumer chooses to purchase this product, they have to check how much is packaged in this bag, the quality of the product, and of course the brand. Again, there is hesitation to purchase of these products. One of the reasons that makes these customers hesitate to purchase the product is the lack of knowledge, which is taking the lead there for 4%. So, once they purchase these products, you'll find that the issuance of the technical data sheet of the end product is at a no of 22%. This means that there is no 100% issuance of a technical data sheet on how to use the products, the dry mix mortar products. Again, most of the failures associated with these dry mix mortars, there is high performance. This means there is limited complaint. As you can see, that 96.8% that once they use, for those who have consumed the dry mix mortars, there is limited or there is no complaints on the same. A report from the standard bodies, which were mainly true that the KEBS and the NCA, they agree that there is a coverage of these dry mix mortars in the standards. And again, there is need for artisan training because they are the end users of these products. And there is also need to do joint academic research, government support, and private sector partnership in driving the adoption of these dry mix mortars. To my conclusion is that the dry mix mortar is currently being used by the consumers at 50% rate. Dry mix mortars manufacturers do not adhere strictly to manufacturing codes. This is associated with the failures as evident in the result. Then there is major drivers for adoption of dry mix mortars at the quality of the product. There is need for artisan training and intensified training. There is need for proper regulation and monitoring of dry mix mortar manufacturing. My recommendation is that there should be an intensified artisan training, enhanced research work on dry mix mortars, and there is need to regulate the manufacturers of dry mix mortars, and then provision of the technical data sheet for every dry mix mortar that is produced. Thank you.
Thank you so much. Let's start 20 seconds to go. So, that is Douglas. Yes, then we shall have our gentleman here. Who else will be, then I'll be closing. Anyone else pending? After him, it should be the last one, the second last. We did not have Lena McKenna, and we also don't have Livingston Jail. So, who are not present, so you will be the last one. Mr. Roque, ujibu maswali.
Niulize maswali.
Na pia waulize na wajibu.
Good evening. I'm Douglas Mwenda, a master's student pursuing Master of Science in Applied Mathematics. The topic is modeling numeric transmission incorporating asymptomatic component and media awareness. My supervisors are Dr. Grace Kaki and Dr. Stephen Karanja. So I'll start by defining the terms. So asymptomatic component, asymptomatic is the ability to spread a disease without showing symptoms. And then modeling is a way of representing problems mathematically. So pneumonia is an acute respiratory infection that inflames the alveoli in the lungs. That is according to WHO 2022, and you can see the symptoms. And the research shows that the disease is able to spread through asymptomatic ways and symptomatic ways. Symptomatic are those people who show symptoms. A statement of the problem. So data from global burden of disease shows that over the recent years, pneumonia cases have been increasing. You can see also the cases in Kenya, and we have data from Mayo Teaching and Referral Hospital back in 2022 that shows some screened samples from people visiting the hospital showed that at least 12.3% were infected with the pneumonia disease. Then the specific objectives. The first objective is to formulate a mathematical model incorporating the asymptomatic component and awareness. And secondly, we have to determine the existence and bit of equilibrium points, bifurcation and sensitivity analysis of the model. And then number 3, we have to assess the impact of effective mean awareness on pneumonia transmission involving the asymptomatic component. So this is the model flowchart. So the entire the disease has been divided into 7 compartments. The second compartment shows different progression stages of the disease, from susceptible, S for susceptible, E for exposed, IS for infectious symptomatic, IA for infectious asymptomatic, then TS is treated asymptomatic, TA is treated asymptomatic, and then we have R, which is covered. So on objective 2, we perform sensitivity analysis. So this is where we see how each parameter contributes to the reproduction number. The reproduction number in mathematics tells us that if one person is infected, how many people are likely to be infected with the disease from that person. So if the reproduction number is 2, that means that person will infect 2. If it's 3, then the person will infect 3. So these are the parameters. The parameters that are positive shows that if the parameters increase, the one that you can see 1, 1, 1, those parameters are linear. They have linear relationship with the reproduction number, which means a 10% increase in that parameter will range to 10% increase in the reproduction number. Then we have other positive. Also, when you increase those positive parameters, then the pneumonia disease increases in the population. Then you have the negative parameters. This shows that control effort should seek to increase the negative parameters in this screening and treatment. Then on objective 3, we did numerical simulations using MATLAB software. I don't know why this one is not showing entirely, but I can still discuss it. So you can see we have 3 lines. We have the blue line, orange and green. So row of the parameter that looks like P represents effective mean awareness. On the y-axis, we have the reproduction number. The y-axis is the vertical. Go back again. Again. Okay. Yes. That one, thank you. Sorry. I can still discuss it, it's okay. So you can see the first one, the blue line, we have reached effectiveness at 0.2, and you can see we have higher reproduction number. Then we have the orange middle. Then we have at higher effectiveness, we can see we have low reproduction number. Then as awareness increases, more people are going to get screened and receive treatment, which means over time the reproduction number will reduce and go to below 1. When the reproduction number goes to below 1, that means if you infected the disease, you infect less than 1 person, which means the disease cannot be sustained in the population. The disease will finally die out. Then we have this graph. which shows the effect of the asymptomatic transmission of the reproduction number. So the x-axis, we try to vary the transmission rate of asymptomatic. So you can see here from 0 to 3. So if the transmission rate of asymptomatic is 1, that means one person has ability to infect more than one person, and you can see this one has a linear relationship with the reproduction number. As these people become more infectious without being noticed, we have more sick people in the population. Then we have this graph. This is a graph of combination of strategies. We have combination of behavioral, self-protective behaviors like wearing of mask and self-isolation, and then we have medical screening. And you can see, for the disease to be eliminated from the population, we don't only need self-isolation or wearing of mask when we suspect exposure. We also need to get screened and receive treatment. Conclusion. So, a combination of behavioral isolation and medical screening is effective in reducing the reproduction number to below 1. And then awareness can be an effective tool in reducing asymptomatic individuals in the population, because these are people who receive, who don't show symptoms, and therefore they continue spreading the disease without knowing. But when they know, they are likely to get screened and receive treatment. Then recommendation, we strengthen screening programs to detect asymptomatic cases early, especially in high-risk setting. And then we have public health efforts should protect both media awareness effectiveness and screening interventions to effectively suppress disease transmission. And finally, we integrate awareness strategies into public health policies for pneumonia prevention and control. Thank you.
So much. Let's clap for him. Our last speaker, please.
Good evening. I'm Kennedy or Diabo Angila, MSc Environmental Science. My supervisors are Professor John Moria, Dr. Nicholas Mwenda, and Dr. David Bab. So I'm doing a presentation on sustainable e-waste management for urban centers, challenges and opportunities in Meru municipality of Kenya. Introduction. So waste electric and electronic equipment, that is WE, is an electrical stroke electronic equipment in its end of life or end of use. Globally, e-waste reached a record 62 metric tons by 2022, and short life cycles and near obsolete imports of these electronics accelerate rapid device accumulation, and of course, leaching of these heavy metals cause critical environmental and neurological damage. And Kenya's law recycling states NEMA mandated EPR, of course, according to WISREC 2024. So the problem statement: urbanization and technological growth accelerate local e-waste generation. Secondly, hazardous components within these electronic devices jeopardize or limit or cause a barrier to environmental integrity and public health. Informal management systems bypass established institutional and regulatory frameworks. And lastly, empirical baseline data on generation and disposal of e-waste remains deficient. Now, a different methodology. I used both qualitative and quantitative track design, of course, that is through the use of questionnaires and interviews. My study area was Meru municipality. The sampling technique was both stratified and simple random sampling. The sample size was 400 questionnaire respondents and 4 key interviews. The target population were households, educational institutions, private businesses, government offices, medical facilities, informal recyclers, and repair workshops. For validation and reliability of my data collection instruments, Cronspus alpha test yielded a 0.7 result for my pilot study at Chuka Township. I used, of course, I administered 400 questionnaires, and out of these, I collected